Okay, well, welcome back. This is the last meeting of Engineering 443, 543 virtual tissues. You guys get to do the presentations today.
Just like last time, we're going to start. Logan's going to give you guys some background, and then I'll talk about the model, and then we'll get to some pretty cool results.
All right, so I'm going to kick off today by just going over the background for quorum sensing again. So as you may remember, bacteria are able to coordinate population level behaviors using this communication system we refer to as quorum sensing. And quorum just refers to like as a necessary group. So like in the government, we use the word quorum to describe like the necessary number of representatives for a voting group. It's the similar kind of concept here. So bacterial cells are constantly excreting these molecules called autoinducers. They're metabolic byproducts. So as the cell goes through like different amino acid biosynthesis pathways or sugar metabolism pathways, they'll create these molecules that are then released into the environment. And the accumulation of these signals in the environment is directly tied to population density because of this. And bacteria, as we all know, have a large set of behaviors. When they're alone, they want to be doing certain things like looking for nutrients, looking for surfaces to attach to, trying to get inside of a host, things like that. Versus when they're in a group, there's a different set of things they want to be doing, like acting virulent, releasing from the biofilm, getting away from nutrient poor areas. And these behaviors, the high cell density behaviors, the ones where they're all together, are really energetically costly for the bacteria. So they only want to turn them on when they're necessary. And quorum sensing allows them to be phenotypically flexible in this way. So they're only going to turn high cell density genes on when there are lots of them around. And they're doing this by sensing these signal molecules that are being excreted. And so after they search or hit a certain threshold of these molecules, they'll switch on these high cell density genes and switch off the low cell density genes. And when that threshold falls, or the molecules fall back below that threshold, they turn off the high cell density genes and back on the low cell density ones. And this is really important because in a lot of human pathogens, this process regulates over 500 different genes. And almost all human pathogens that utilize quorum sensing regulate their virulence in this way. So like cholera, for instance, when it makes you sick, it utilizes this gene regulation program to kind of facilitate its pathogenesis. And some of you might have heard of necrotizing fasciitis. I didn't include pictures for obvious reasons, but it also regulates its pathogenesis in this way. And there are tons of other ones like Staph aureus, the stuff that causes cavities and periodontal disease, they all regulate those phenotypes using this. So obviously, it has become a large target for research in terms of biocontrol and therapeutics, because not only does it cost us a lot in terms of our human health, but also in terms of our agriculture. So like fish and shrimp farms, oyster farms, it costs them billions of dollars a year in livestock damage, but also in terms of our mammalian livestock as well. And the cool thing about this is it also regulates what we call the production of a public good. So when the bacteria are in nutrient complex environments where they wouldn't normally be able to eat the things present, they release these public goods to kind of chew that stuff up to make it easier to digest. And that is a quorum sensing regulated phenotype. And I bring that up because that'll be important for understanding our model a little bit later. And Nick, if you could go on to the next slide for me. So when we think about quorum sensing, we aren't typically talking about a homogeneous population. So typically bacteria exist in very heterogeneous populations, and that can be both in terms of different species, so like your microbiome, or same strain, same species, but different phenotypes. And in terms of quorum sensing, we can have what are called unconditional cooperators, so cells that for some reason just get stuck quorum sensing all the time. They have always their high cell density genes on, they're always pouring energy into those pathways. Then we can also get cells that are called defectors, where they decide, I am no longer contributing to the public good. I am just going to reap the benefit of all the other cells. And the cool thing about these cheaters is that actually on a cell-to-cell basis, so when we're looking one cell to one cell, the cheaters always have a fitness advantage over any other quorum sensing phenotype. The cooler thing is that on a population level, wild type cells always win out over cheaters. They always, because of the greater phenotypic flexibility, are able to outcompete cheaters in dynamic environments, which is really cool. So this is an evolutionary example of something that might be beneficial for a single cell, but in terms of a whole cellular population is not. The interesting part about this is that we don't understand how defectors and unconditional cooperators impact quorum sensing based drugs. So drugs that are targeting quorum sensing systems to shut virulence off, and how they impact infective outcomes and the overall fitness of bacterial populations. Because if they were greater in fitness overall, obviously, bugs would evolve towards totally defector or totally cooperating strains, but they haven't. So this model seeks to kind of understand like why or how that's happening. And Nick, if you could go to the next slide, I'll talk about the modeling. So in terms of quorum sensing, it's not new to think about modeling quorum sensing. That's been done a lot, but typically those have been limited to like fitness landscapes. So understanding how or why quorum sensing evolved, and then also just how those populations adapt and change in their makeup over time. So the first model we're going to talk about is from Melky et al. It connects the production of those signaling molecules to growth to kind of simulate a public good. And we don't necessarily do this directly. We just kind of, for some of the concepts Nick will get into when he talks about our architecture, we just use this as a good basis to be like, okay, these people have connected signals to different quorum sensing phenotypes and had great results. So we kind of knew that that was a positive direction for us to head in as well. And then the Sarzan et al. Model used stochastic instead of fitness landscape-based mutation to kind of look at how quorum sensing populations change over time in a given environment. And because their model worked really well for what they were looking at, we decided to adopt the same evolution scheme. So you'll notice in our architecture, we use a stochastic mutation switch rather than a fitness landscape. And then we have a wealth of in vivo data from my lab and then other labs as well, that show how cheating impacts the evolution of quorum sensing regulated cooperation. So if you look at the panels over here, you'll see different shades of gray and black. Those gray indicate the appearance of defector strains. So that's the fraction of the total population that are defecting. They're not quorum sensing, right? The black is the cooperator strains. And you notice that in 11 of the 12 scenarios, wild type cells eventually win out. So even if they initially start to lose, they'll pop back or they'll persist even if they remain a minority. And this is actually really special because in these scenarios, they're actually set up in an environment to favor the defector cells. And we'll see that quite often the wild type is still able to overcome them, which is really cool. It demonstrates that phenomenon I was talking about earlier. But the nice part about this in vivo data is that it informs the model we're building and kind of gives us different health checkpoints to make sure that what we're doing is actually relevant to real life, which is obviously the goal of all of our models.
Okay, so I'll take over here and just kind of talk about how our model came together, starting with the ideas from the papers and then some of the additional things that Logan added, or Logan and I kind of dreamed up together. So starting with the impact, quorum sensing inhibitors. These are drugs that inhibit this quorum sensing genetic response. Are being researched, they're being developed, as Logan mentioned, since virulence is often a quorum sensing regulated behavior, it could be useful drugs for combating that. And then just looking at a model of these different phenotypes, like this diverse population, and how, given a quorum sensing inhibitor, how the phenotypic kind of proportions between the different populations, like what their fitness landscape looks like. And then, yeah, looking at cheating versus unconditional cooperating. So it's two different atypical cell types we wanted to include. So that actually came from the Cesarin, or however you say this name. What he did in his model, using a lattice-based model, just like CompuCell, but they didn't say the name of it. They use different alleles, and then each cell would have two alleles, and that would regulate how they behaved in the environment. And then this Melky et al. Was the other paper that we wanted to kind of take an idea from. And what they did is they modeled quorum sensing, but instead of having the cells produce a public good, which in turn affected their nutrient intake, which is what me and Logan did. They just made a mathematical equation to say, okay, we have this population density. Theoretically, there'd be this much public good that would influence their metabolic energy in this way. But we really wanted to make it a chemical field in CompuCell. And then you'll see more in the results section, but we might mention a couple times here, we're using data from Logan's work in the Van Kessel lab to compare our results to the wet lab results. So starting with our assumptions, we're going to talk about the complex nutrient as one of the chemical fields in our model, and the uptake of this complex nutrient can only be done when the cells are uptaking enough public good, which is in turn produced by quorum sensing active cells. So what we're going to see is our window that watches the complex nutrient uptake is our qualitative way of measuring the quorum sensing behavior that's actually occurring. The second thing that we incorporated just to keep our model kind of in tune with biology is the stringent response. It impacts mostly the defector cell type because these are the cell types that feel the largest effect. When I believe this happens in all three of the phenotypes. But what the stringent response is a response that these, like a behavioral response, these bacterial cells will show when they're low on nutrients. So there's not that much food in their environment. They don't have that much nutrients saved up. So then they kind of down regulate. But of course, regulating these stringent response genes takes in a metabolic effect. And then the last assumption, and a pretty big part of our model, is the metabolic energy, which just represents the resources that's stored by the cell. So it will be used for mitosis. You need enough metabolic energy, obviously, to divide into two cells. Our growth is proportional to how much energy you have saved up. Of course, it takes up metabolic energy as well. And then if you run out, you die. So yes, those are our model assumptions. Just getting to kind of the basics of our architecture. We had 3 cell types, a wild type, and these different cell types are taking place of the phenotypes in the Cesarean model. So the different cell types, they're all the same bacterial cell or what they represent, but the cell types show the different kinds of behaviors. So the wild type produce the public good only if autoducer levels are above a threshold. So quorum sensing is turned on if we have enough cells packed together, and turned off for them if there isn't. The unconditional cooperator, these are the cells that Logan mentioned that are stuck on. Whether there's enough cells or not accumulated, they're always going to produce that public good. And then last is our defector. That's our cheater, you could think about it. They will take in the benefit, but they never produce the public good. So they're like stuck to quorum sensing inactive. The chemical fields we had 5. We had first an autoinducer, which is secreted by all the cells. It ends up being your kind of your analog for the population density. The public good, which is produced by quorum sensing positive cells. We called it a sensor positive in our model. The nutrient was presented in the initial conditions. We did some experiments where we did nutrient flushes, which is something you'll do in the wet lab experiments. So we wanted to incorporate that too. So after every 500 MCS, if this parameter is turned on, the nutrient, which is like it initially starts in every condition. Our initial condition is set to a constant. It will re-establish that concentration. Okay, we have the complex nutrient, which also initially starts in the lattice at every location. And it is not added every 500 MCS. And then last is the quorum sensing inhibitor, which is put in from the X, so the edges of the simulation. And then we had one where there's a linear gradient. We ran some experiments that way, and then we ran somewhere the gradient was coming in from both directions. And then the way we can kind of turn this on and off is with a parameter that doesn't take away the field, but it turns off the interaction that this would have with the cells inside the Python for the code. So yes, the metabolic energy. This was a dictionary attribute for each cell. Like I said earlier, it kind of represents the stored up resources. So if there's no resources stored up, the cell dies. If the cell does not have a lot saved up, but we're not dead, our mutation increases. This is a biological phenomenon that we see with bacteria. If they're starving, the chance of mutation increases. Next, there's an upper ceiling. So just to keep our metabolic energy values from skyrocketing, we put a ceiling at 2000. If your metabolic energy is over 200, you can mitosis, as long as you have a big enough volume also. And then the metabolic energy is modified by these three things in this equation. So nutrient uptake, how much nutrient did you uptake? There's a decay rate, which is constant, a growth factor, which is based on the metabolic energy, but we'll show that later. And then a decay modifier. So this is how we kind of really differentiated the different cell types, is with this decay modifier. So just in our update equation, you can see here's our total nutrient, adds to the metabolic energy, and then these three subtract the constant decay, the factor for how much we grew that MCS, and then the unique metabolic modifier. I mentioned these a little bit here. We have the wild type cells, which have a decay, so an increase to the decay when they are actively quorum sensing. As Logan said, these genes on an individual basis are more expensive, so we wanted to incorporate that. The unconditional cooperator cells, on top of that active quorum sensing, also have an early quorum sensing decay. So now, since they're always quorum sensing even before there's a metabolic improvement, like a justification too, you could say, these cells, they're always quorum sensing. So we put in an additional penalty to the metabolic energy where it decays even faster if there aren't other cells that are secreting this autoinducer around them. And then the defector cells, this was the stringent response I mentioned earlier. I'll show the equation for that. But if nutrient is readily available and the population density is high, we have a larger decay. But we'll get into that in this slide. So here's our three decay modifiers that I was just talking about. So if actively quorum sensing, the wild type cells have a constant decay. That just says you're quorum sensing. We need to subtract resources for that. Then we have the unconditional cooperator. So we have this threshold, an autoinducer threshold that says when this autoinducer threshold is reached, quorum sensing now has a metabolic benefit. Before that, we add this extra metabolic cost to the unconditional cooperators. We used a log 10 equation that was really just fit to the behavior we wanted to see. Yeah, and then with the defector, there is a metabolic consequence. So our stringent response, if there was low nutrient, so nutrients on the bottom of our fraction, and on autoinducer is low as well. So not a lot of nutrient, you're hungry, not a lot of autoinducer to add in that public benefit. So yes, those are the extra decay modifiers for our different cell types. Now the growth factor. So this also was added to the negative parentheses from our metabolic energy. This just determines how much the cell grows. So it will be added every MCS, the growth factor, to the cell's target volume. And then the way that we calculate it is here. Just we tried out a few different coefficients for that and just found 0.001 to work best for our results. And then there's a starvation bonus. So you would think if the cells are starving, so they have less than 500 resources stored up, the nutrient uptake is also very small. There's not a lot of resources. The cells now decrease the growth factor 10%, so they grow less, and then in turn that will take less out of their metabolic energy. So they're kind of prioritizing life over growth in this case. Secretion. Autoinducer is secreted by every cell, every MCS. This was definitely our variable for measuring population density. The public good was secreted by sensor positive cells, so all the unconditional cooperators, and then the wild type that are turned on. The nutrient provided by the simulation boundaries, also in the initial conditions, it was present in the whole lattice, and then the complex nutrient uptaken uptook by every cell, but only ones that have this public good uptake that comes from quorum sensing. Yes, so only if you have public good, that's what this part is. So the drug, so the quorum sensing inhibiting drug that we were modeling here is called PTSP. And what it does in our simulation and also in real life biology is it decreases the secretion of a public good. The IC50 refers to the concentration at which PTSP has 50% of its inhibitory effect, the concentration of 50%. Is how I remember it. And for PTSP, it's 0.35 micromoles. And here we modeled that. So 0.05 is the output of our equation. If our X value, our uptaken QSID is 0.35. So then we get 0.5, and this is put into a proportion for the excretion of the public good. So it's capped at 1. You can't secrete more than 100% of your public good secretion rate, and then we cut it in half here. And then just some other aspects that we included just to make it biologically accurate. We have mutation, like Logan mentioned, we had stochastic mutation. It started after 500 MCS just to let our cells kind of equilibrate, get our chemical fields in. There's stochastic mutation, and if their metabolic energy is low, there's a higher chance of mutation, which was modeled after this Cesarin paper. And then chemotaxis, all cells chemotax towards both the nutrient and the complex nutrient, but if complex nutrient uptake was high, it was favored. So if quorum sensing was turned on, we wanted this complex nutrient, but that also meant that the amount of nutrient was likely low. Yeah, and if nutrients decrease, is what I just said. So yes, now we're going to transfer over to Logan to talk a little bit about our.
Yeah, so after we said about like with this architecture, we wanted to really like understand what our model is actually going to produce for us. So we separated this into two bins, right? We have our quantitative and our qualitative outputs. Qualitatively, it allows us to kind of see on a small scale what the spatial structure of the allelic populations are. So, we wanted to see, are we seeing an invasion of the defector cells into clumps of quorum sensing positive cells? So the UC or unconditional cooperator and wild type cells, do they kind of wedge their way into these populations to get access to that public good? We also wanted to understand how the nutrient field was shaped and changed over time, and if that played into like how the allelic populations were structured. So being able to look at both was a great way for us to be able to do this. And then we also wanted to see how different drug fields affected this population structure and the overall virulence of the population. And that in a qualitative sense, we just mean like by looking at public good excretion, we can kind of start to understand how well these drugs are shutting off the quorum sensing behaviors, right? We'll get into the results here in a second, but you'll hopefully notice that like the amount, because the colors change, the maximum amount gets decreased quite significantly. And this was a great way for us to kind of visualize that. And then quantitatively, to kind of mimic the in vivo data we had been seeing, we wanted plots describing population size over time, and going hand in hand with that, we wanted also the fraction of the population that's actively cooperating, just meaning that they are quorum sensing capable. So it says fraction of the population cooperating. That doesn't necessarily mean they're quorum sensing. It just means that like they're either the wild type or unconditional, because those are considered cooperating cells. And then we wanted to understand like how the number of active quorum sensing changed over time. So S state just refers to whether or not the sensor is triggered. So they're seeing enough autoinducer to quorum sense. And we wanted to see how that changed over time, as well as how much energy each cell possessed over time. So were there certain events that we could see in these quantitative plots that kind of were then mirrored in the qualitative ones as well. Nick, if you could pop over to the next one for me. So we had some learning objectives for this. We want to see, just to make sure our model is working, that the autoinducers accumulate in a population density dependent manner. So we want to show that this AI field accumulates differently under different population densities, meaning like if they're really close together, all of those cells are clumped, so there's lots of autoinducer present around the cells, that's high. But if they're spread out, there's less. If there's a ton of cells, there's more. And that kind of tells us that the autoinducer field is in fact like a good way to kind of look at population density, because your population can be really dense, and in reality bacteria do sometimes, even if there's a small number, they'll clump to artificially increase density. So we wanted to be able to capitulate that here and make sure that we were actually measuring phenomena like that if they were happening. And then we also wanted to understand that demonstrate that complex nutrient concentration was inversely proportional to the concentration of public good. So as public good rises, that complex nutrient should decrease. And the cool thing we really wanted to emulate was a delay. So as it rises, after a short delay, we then see the decrease. And that's because bacteria can't instantly switch genes on. It takes time to not only turn the genes on, but to get the mRNA and to translate that into protein, and to have those proteins actually be conveying their activity. So being able to capitulate that delay shows us that the way we've coded it kind of really is starting to mimic that biology. And then we also wanted to show that the inhibitor molecules significantly decrease the activity of quorum sensing behavior. So we show this by the public good, like I said, should be cranked way down. And we should see that in presence of high drug, there is no public good. And if there is any, it's being expressed where there's very little drug, which we get into with our gradient experiments. So getting to that, we want to understand how different gradient distributions impact the inhibitor and its effectiveness, and how different IC50s, as well as different ways to model drug activity, begin to affect what the drugs are looking like on the population level. And then just to make sure users were understanding, we have a steering window with all these parameters. In our learning materials, we include in the foreword just a quick explanation what each learning parameter does. And just in case we have some people who aren't as familiar with coding, we also include explanations for things like 1 is true, 0 is false, things like that to help kind of make it more approachable for everyone, which I will show in a little bit. Actually, Nick, if you want to pop over to the learning materials, I can do that right now before we get started. So we start off with that forward, like I mentioned, we just walk through each of the parameters. For the sake of time, I'm not going to do that here, especially because we're not going to get into all of them in our presentation. But if Nick scrolls down, you can kind of start to see we have quite a few parameters, but each of them has a description that's sufficient enough for someone who's familiar enough with biology to understand what they do. And I did test this with a population of my lab mates who have no clue how to code. So it is apparently enough for people to understand what they're doing. And if you scroll down to section 1, Nick, so that first learning objective is to demonstrate that our model is capable of doing this autoinducer accumulates in a population density dependent manner. So I'm a really big fan of not just showing or not just telling, but showing. So I have thinking exercises in the beginning of each one. So Nick and I thought it was really important that when users approach the model, they understand the biology behind it, because every quorum sensing system is a little different. And for people who do just modeling, there might not be that as in depth of understanding of the biology behind it. So we have people kind of hypothesize about things. Obviously, they don't have to, but just even getting them thinking about it as they approach it, we thought would be beneficial. And then I just walked them through the exact paths and settings we wanted. You can see some results, which I'll get into in a second, but I give them examples of what things should look like. And then at the bottom of the exercise, I have what different results they should expect to see based on different parameters. Just a quick note about for this specific example, some of the settings will cause a division by 0 error. That's normal depending on how they're playing with it. I wanted to make sure that they understood that that could happen. And then just spaced note parameters that they find interesting or useful down below. So that way they had a place to keep record of it. And then in the next one, we talk about that relationship again. I walk through the background. So for those of you that don't know, like complex nutrient, that's not just like a made up thing that Nick and I had. Their bacteria in real life, like I said, will actually use this phenomena to eat complicated sugars. So I walk through that a little bit. And then same deal, different parameters, examples of results at different time points, ideas. And I go through that for the inhibitor drug as well. Nick, if you want to scroll down to section 3, I believe it is. Yeah, section 3 for me. And then if you want to scroll all the way to the bottom for that one too, I walk them through exactly how to change the IC50 parameter, because that is not, we're still working on a way to kind of get that working in the steering window so they can just type in the value and it automatically changes the equation. And I wanted people to know, so a lot of people will use sigmoidal functions for inhibition. We used a log function for this because of the way we were looking at things, but we're like working on adding in the sigmoid option as well to toggle on and off for different types of drugs that's relevant for different reasons. For what we were looking at, it wasn't as big of a deal because we never dealt with really low concentrations of the drug in terms of like really trying to understand that. Because our IC50 is so low. And for troubleshooting purposes, we just put a crap ton of drug on there to make sure everything was happening correctly. But the sigmoidal curve comes in useful like when we start to do more heavy duty experiments of the like lower end of the gradient and how like that affects evolution over time. But because in my lab, we currently use a higher concentration of the drug, we stuck with the logarithmic function for that. And then also some labs favor that for certain things anyway. So having both was something we thought was important. And then if you could go back to the slides, Nick. So that was like just a quick overview of our educational materials, but all of them the same concept. It's the biological background, a few quick questions to probe, like, get them thinking, exact walkthrough of how to change the settings and a basic setup, and then expected results in places for them to know cool things they see. All right, next slide, please, Nick. So as we talked about the last time we did our presentation, the really exciting thing for us was we were able to really quickly capitulate exactly what I see in the lab. So on the left, you'll see what's called a light curve. The y-axis is the bioluminescent units over the cellular density. So how many cells there are versus how many light is produced. So it's controlled for different cell populations. And then the x-axis is the total cell density. And what you can see is that we start at this initial puncta, which kind of represents the cells grow really high, and I back dilute them. So they're still taking the time to shut those genes off. And so we get this U-shaped curve. And as the cells grow more, they turn that behavior back on and begin to bioluminesce again. If you look at the right, we were able to successfully mimic that in the simulation. So you can see when the simulation starts, the cells are really close. So there's lots of autoinducer uptake. There's that puncta. We see the in vivo data. It goes back down as they spread back out, and it rises steadily again as the cell population grows. And so that was really exciting. This was the first big checkpoint and something we wanted to make sure we had down right before we continued. And like I said, like, because I literally did this experiment as we were building the model, we know this isn't just like some fluke. We were able to exactly see what we see in our model in real life as well. And then Nick, if you want to go to the next slide for me. Next was that inverse relationship I was talking about. So you can see four different fields here. Top left, we have complex nutrient. Top right, we have the public good. Top or bottom left, the sensor. So when you see cells turn red, that means they become quorum sensing active. And then in the bottom right, we have the cell field. And what you'll notice over time is you see lots of red. What I really want to draw your attention to is the max and minimum numbers. You'll see as that max number starts to decline. When the cell population gets really dense, it'll start to get around the 70s and then north of 100, and you'll then see the complex nutrient begin to decrease. That lag has to do with the gene turning on, like we mentioned, but then also because they don't really start to utilize those complex nutrients until they've consumed all the simpler ones. You can see now the cells are turning on as they reach the quorum sensing threshold. lots more public good is being excreted, and you'll see it quickly climb from just below 1 to, well above into the 10. So it's an order of magnitude increase quite quickly. And then over time, it'll start to reach a second order of magnitude increase from the initial value. And you can see the minimum value. I'm so sorry, I have to take a drink. My throat just. You can see the minimum value in the complex nutrient field begins to decrease, and that yellow appears in the field as they consume that nutrient. And we see that the public good has climbed into the next order of magnitude, and that delay was successfully mimicked as well. So that was really exciting. A more complex behavior, and we were still able to mimic that on this scale, which was really exciting. This was one of the biggest hurdles I was nervous for in terms of us being able to mimic it. But we were able to successfully do that. So that was really fun. And then if you want to exit out of the video, Nick, and move to the next slide for me. So this was another really big hurdle. This was in vivo data that wasn't from my lab. So this was completely out of my control. I had to call the person and kind of talk to them about what they did and what they were seeing. And we wanted to recapitulate this because this was, we started with a really simple population density dependent, and then a little more complex. This takes it the next step up in terms of emergent and complex behaviors. We wanted to see if we could get defector cells to invade from rare when we kind of pushed the environmental conditions to favor that cell type. And so we used Eric Brugger's study from the Waters lab at MSU to kind of set the bounds for this. He's a really good quorum sensing evolutionary expert, and my lab has a close relationship with his lab, so it was really nice to get to work with him on this. But basically what we did was Eric took multiple generations of cells. And when I say multiple, I mean like several thousand. And he passaged them in conditions that favored the defector cell. And there's a lot of complicated metabolism in understanding what situations favor defector cells. The long or short of it, just so we don't, I know not everyone's a biologist, but basically, defector cells are really great at eating that simple nutrient. So if you constantly wash in simple nutrient, those cells will begin to take over the population because quorum sensing behaviors never become important for energy uptake. And that's kind of like in terms of what I was talking about earlier, where individual cells, defector cells always beat out the wild type or quorum sensing cells. This kind of takes that phenotype and expands it to the population level rather than keeping it limited to individuals. And that phenomenon is still, we understand it more than we did when Eric initially published, but it's still a field of like really rapid study. So being able to mimic something we don't fully understand was a really big health checkpoint. So if we could mimic that without fully understanding it, that was something that would tell us that our model is doing a pretty good job at mimicking what we see in vivo. And you can see here that we were able to successfully create a defector invasion from rare. So we started a population that is 96% cooperators. And we do just a few, in terms of our simulation, it's about 10 generations. And within 10 generations, the cooperator cells drop to below 10% of the total population, and then defector cells rise above 90. So we have the defector cells growing to completely dominate the population on timelines that are similar to what Eric was saying. Nick, if you click, it'll pop in one of Eric's graphs for us. So this was the defector invasion from rare. Just a reminder, the black is the cooperator, the gray is the fraction of defector cells. This looks a lot more tiny because he was doing 2,000, we did 10 just for timeline purposes. But you can see down in the bottom left-hand corner, that's when the population of cooperator cells drops to around 10%, just below 10. And that's what we were able to successfully mimic. So that was really exciting. This is a behavior that we understand, but we don't understand the mechanistic side of it. And we were still able to kind of demonstrate that our model was capable of reproducing this behavior. So that was a really, really exciting milestone for us in terms of development. And after we passed all these health checkpoints, we decided to move on into an area of increasing unknown, which was the inhibitors. So Nick, if you could pop to the next one for me. So I have 3 different videos here. We'll start off with no active inhibitor, which is a similar video to before. But this time we have a different cell field. So we have the complex and public good on top like normal, but now we have a metabolic energy plot in the bottom left-hand corner, replacing the sensor field we had before. You can see the green lines early on will kind of start to plop down. They're incurring that early quorum sensing penalty Nick mentioned. The metabolism behind that is that those cells, because they're not phenotypically flexible and able to go low or high, they lack certain metabolic shunts that wild type and defector cells possess, which makes them incur greater energy costs. So we can see that those cells pretty early on are lower in energy compared to the wild type and defector. And then in the red lines, I hope you notice these kind of humps that are forming. That is a consequence of the autoinducer field building up. So they're incurring that stringent response consequence that Nick was talking about, which prevents them from really regulating that response very well and adapting to more stringent conditions like the wild type cells are. And you'll notice that those cells' energy decline is more of kind of like a slow logarithmic slope down. They're able to handle restrictive conditions better. And what you'll notice is that late in the simulation, the wild type becomes the only persistent cell population. And if we let this run, because they start to fall below mutation parameters, you'll start to see certain cell types pop in. I don't know if this happens in this video, but when we do our experiments in real time, we do start to see new ones pop in, but those cells are quickly selected against, and wild type continues to persist as the major population. In this next simulation, we have a constant inhibitor field of what's equivalent to 10 micromolar. So this is a whole order of magnitude above the IC50. This should completely ameliorate any quorum sensing phenotype. You'll notice that we still get low levels of public good, but that maximum value is going to stay below 1. So instead of being at the 100 when we reach a total quorum sensing population, we'll have decreased it by 3 orders of magnitude to below 1, which was really exciting. Sometimes in certain simulations, you'll see it fluctuate a little above, but it never goes far above 1 if it ever reaches that point. It just depends on how the cells are clumped and how, because they're uptaking it at the same time, there's some of the dynamics there as well. But the production stays very low in this constant field. And what you'll notice about this is that the wild type cells also begin to incur that same hump shape that the defector cells do, because they are no longer able to correctly regulate that stringent response Nick and I have talked about today. And the unconditional cooperator cells, they just do quite poorly in general, because now they're spending all this energy to excrete this public good, and it's literally reaping no benefit. And you can see in the complex nutrient, there is some being eaten. That just represents the fact that no drug is perfect. This is what's known as a competitive inhibitor. So it's binding the same ligand pocket as the natural ligand. And the enzyme kinetics of that just say that there's never 100% repression, because things associate on and off. So we will see low levels of digestion, but late into the simulation field, we still see that it's not really decreased much at all, which was really cool. So we were able to correctly mimic what we see in the lab. I don't have graphs of in vivo data here just because that drug is still in development, but it's pretty close to the proteolysis assays we observe, as well as the bioluminescence curve, which was really exciting. So the next thing we wanted to test was an inhibitor gradient. So this gradient is from left to right. So at the X minimum is where the concentration of drug is the highest, and that steadily decreases as we reach the X maximum. And you'll start to notice that this time we see an increased expression of that public good and an increased digestion of the complex nutrient. And that changes our energy dynamics, which you'll be able to look at in the metabolic energy plot, as well as different spatial distributions of the cells as well. So you'll kind of start to notice that cells towards the minimum field, especially if they're quorum sensing capable, tend to do a little better as far as persistence goes. And you can see that this maximum value for the public good has already increased above 1, but it's still staying below 10. So we took what was a 3 order of magnitude decrease, and it's just now a 2 order of magnitude decrease. And that is coming from, you can see the tight cluster on the right-hand side of the field. So the X maximum, where the drug is lowest, which is really cool. So we are not seeing very much excretion of public good where the drug is highest, and we're seeing a lot more where it's pretty much non-existent, which tells us again that the dynamics of our simulation are mimicking exactly what we would expect. And you'll start to see those cells that are quorum sensing will persist a little longer, and then they'll begin to invade into the area where the drug is low, and they can eat that complex nutrient, which you're seeing as the yellow cloud appearing in the top left-hand side. Yeah. So that was really exciting. The thing that's most interesting for my thesis is understanding how these drugs actually work in an infection, right? When we use antibiotics, the thing we didn't consider was the quick development of resistance. So when Fleming first found penicillin, we just used it, and within a few years, it became pretty much useless, right? And so the important part for me is understanding how that might happen to quorum sensing drugs before we begin to use them so we can get ahead of the problem. And the way I do this is, instead of nominally using just like high nutrients like a test tube, like all of the experiments have, I did what's called nutrient cycling, where Nick was talking about washing in new nutrients every few hundred MCS. What this does is simulate the entrance and exit from a host. So hosts are considered nutrient rich. There's lots of stuff that's available for the bacteria to eat. And then the external, like planktonic environment, is nutrient poor. So like in the case of the bacteria I study, when it's inside a fish, it can gobble, gobble, gobble all of that good food. And when it gets released from the fish to find a new host, the seawater is what's called a nutrient poor environment. And this is kind of where we understand quorum sensing to be really beneficial. So I talked about that phenotypic flexibility. And this allows them to be phenotypically flexible. As they go from high to low, they're able to change their gene regulation to conserve energy. And when they go from low to high, they're able to more quickly turn on those genes so they can get as much energy as possible before they get kicked out again. And so I did these cycles with and without inhibitor. And you'll notice that in both cases, wild type cells still persist. Now, this was in contrary to my hypothesis, where I assumed that the wild type cells would not do better with the inhibitor present. I was assuming that either the defector cells would do better, or that it would remain a mixed population. So this was really cool. This was counter to what I expected, and it was counter to what my mentor and some of my colleagues expected to see as well. And the cool part is I have a few undergrad mentees. They are currently working on actual in vivo experiments to see if we still see this same thing within the organism. And I'll get into here in the next slide about like why we might not, and where those errors might come from. But also, it was just cool in general. Like, so far, the simulation had followed perfectly our expectations, and we're looking again at an emergent behavior, and suddenly, it's kind of making us question things. But this phenotype is also being supported by in vitro, so in the test tube data from one of my colleagues in the lab who's studying the link between metabolism and quorum sensing. And I think we got a question in the chat. So, James asked in the chat if there was a general rule of thumb in the evolution that spatial gradients tend to drive rapid evolution, because individuals at the boundary between survival and death can occupy new niches and do just a little better. And this tends to be true of resistance phenotypes. So like in antibiotic resistance, when they exist on the gradient between life and death, they can invade a niche and do a little bit better, and that just propagates over time. And which is why you have to hit with a high dose of therapy for certain things. So that's the issue, is in all of the in vivo experiments, no one has been able to, or in vitro, my bad. So within a test tube culture, no one has really been able to create resistance, right? So this kind of gets at an unfortunate part of science. Like, we like to look for the things we're expecting, right? And people, when they're working on these drugs, they like to say, we're resistant to resistance. And so they passage these cells side by side with antibiotics and quorum sensing inhibitor, and they stop their experiment when there is antibiotic resistance in the antibiotic treated population, and say, look, there's no resistance in the quorum sensing inhibitor population. But I think this is partially because all of those experiments that I have read up on tend to be done in only nutrient rich environments. So like the media that the bacteria loves to grow in, and they're not really done in drug gradients. So they add the drug into solution. It's diffused throughout the whole solution because they spin in incubators. And so that was kind of my main gripe with those claims. There hasn't been like really robust evolutionary work looking at the native context for this phenotype. And that's why Nick and I kind of set out to do this simulation, because we wanted to see if we can simulate all these things and we see this, then I know in my in vivo data to continue these assays, or I'm able to test different nutrient or media conditions in order to inform what I'm doing, to see if I can force them to evolve resistance, so we can understand how that might appear like mechanistically in terms of like what gene strategies they use. So this was really cool. You know, it's informing that actively, and we're still working on kind of improving it as we get new metabolic data, and just to make sure that we're not making false conclusions based on the assumptions I'll talk about now. Nick, if you want to pop to the next slide. So there are a few limitations, as I mentioned. So I don't know if any of you remember, but I talked last time about what's called Simpson's paradox. So small populations are vulnerable to what are called false trends relative to what is called a meta population. So in a normal test tube, we're talking about 10 to the 9 cells. So that's billions or sometimes trillions of cells within a given test tube. And in our CompuCell, you know, there's not enough computing power on this planet to like mimic that many cells. So we're limited in our lattice to just a few hundred. So that means that as we do these experiments, we can see trends within our small population that if we were able to do billions and billions of cells might not exist, just because we're looking at such a local population. We can get around this, or attempt to, by doing lots and lots of simulations, and then looking at the trends of all of those put together. So like that would be like if the experimenter in this graph did the blue and red separate, but then looked at the blue and the red data together as without making distinction between the experiments. That's not perfect either, but that's why we use it to inform in vivo experiments and not to necessarily make conclusions, because we tune the model with in vivo data and the in vivo data with the model, and it's kind of like this positive feedback loop. So it's a limitation. It's not necessarily like a death sentence. This is something that a lot of like bacterial population simulations encounter, where they get a few hundred, maybe if they have supercomputers, a few thousand. And we're looking at using Big Red to like help with the population sizes. But then we also have the fact that quorum sensing phenotypes are tuned by multiple regulons. So the stringent response, right? Quorum sensing plays into the regulation of that, but then we have what's called the REL-A SPO-T regulon, which also regulate these things. The same thing is true for like the complex nutrient enzymes. Virulence is regulated by multiple things, like Staphylococcus aureus uses like 8 different regulons to turn on and off its different virulence phenotypes. And so does the bug eye study. So we run into problems where like, sure, if we shut quorum sensing off, like this should shut virulence off, but we may find that the bugs pretty quickly adapt to just use those other regulons. We haven't seen that in the lab yet, but again, most of the experiments that have been done looking at resistance to quorum sensing drugs are kind of limited in terms of they just do nutrient-rich conditions that don't really mimic the cycling of infections. And there haven't been any, to my knowledge, that look at resistance in like murine or mouse infection models. So that's what my lab is doing to kind of fill that gap. And then the import and export of autoinducers and quorum sensing inhibitors is not super well described. The field, for good reason, assumes that autoinducers diffuse through the membrane due to their chemical makeup. But quorum sensing inhibitors, we have no clue if they use like the promiscuous OMP pathway, so the way the cells regulate their internal osmolarity, which is often co-opted by other molecules, or if there's a different type of import mechanism it's using to get in the cell, or if it's freely diffusing. And these things are, again, limitations in terms of like that would change the dynamics of how things are excreted or taken up, and how the inhibition works, because that would affect the intracellular concentration of the drug. But once again, we still are able to see things that we've observed before. So I'm fairly confident that while this is a limitation, it's not necessarily a death sentence to the usefulness of the model.
Okay, and now we can demo our NanoHub page that we gladly have working. So here we're the quorum sensing inhibiting drug simulation on CC3D, or the tool CC3D QSID for a quorum sensing inhibiting drug. I can drop that in the chat really quick. And I don't believe we have too much more than this. We have our simulation right here. We can show the tool very quickly. Just because I see we're beginning to take up a good amount of time. We could also show, so we've implemented our steering window since last time. We'll do a quick tile. The auto-inducer uptake to where we showed the dip earlier. Then we have nutrient cell field, our cooperation frequency, the quorum sensing inhibitor uptake because it's turned on, metabolic energy. So yes, that's our nano hub. That's our, here we also have our exercises in our nano hub. We share. And then our last little piece of our presentation is just our remaining future directions that we thought about doing. The first still being adding elongation to better simulate certain bacterial populations, because we really envisioned this model being useful for many different researchers, and then also including the IC50 flexibility. So yes, that is our presentation. Thank you guys for your time. And we'll now be taking questions.
Great, thank you. Are there any questions? That was a very impressive and thorough presentation, as Gabriel said. It's awesome. Are there any questions, comments? I'm looking forward to trying out the. It's really been.
I have a question. So first of all, thanks for the great presentation. I can see that you have invested a lot of time and effort to this. First question is quite similar to Elmar's. Like, what decides the cell type, like cooperator or the defector cell types, like mutations or something else? And also, is autoinducer kind of stimulate the transcription factor to regulate gene expression, and what kind of gene expression do they lead to?
Okay, so to answer the first part of your question, the mutation rates, you can either start the simulation with different types of cell types, which you set in the XML file, and the explanation for that is in the supporting documents, but they also can mutate. So the way we have the mutation structure set up, when the cell mutates, it flips to a different cell type, and then it adopts the behaviors of that cell type. And I think Nick could probably provide like a more articulate explanation of how that works if you're interested. But as far as the like quorum sensing half goes, the way the autoinducers work is they bind to transmembrane receptors in the inner membrane. So they diffuse from outside the cell into the periplasm of gram-negative cells, and they bind to transmembrane receptors. And when they're bound, they change those transmembrane receptors from kinases, or things that phosphorylate downstream molecules, to phosphatases, or things that eat phosphate up from the system. And when there's phosphate being depleted, there's like a long signaling cascade that involves like 4 different proteins. When that phosphate is depleted from that pathway, the transcription factor called LuxR, or a TET-R protein, is turned on, and that protein acts as both a repressor and an activator for different sets of genes. And that's really the one, when I said like quorum sensing regulates like 500 plus genes, that protein alone is responsible for that number. There's also a low cell density regulator, but because of like experimental limitations and just like generally because it's not a virulence regulator, the lack of like funding and interest in it, it's not as well studied. It's called AFA. It does a lot of the lower cell density stuff, but that one is turned on in the presence of phosphate in the pathway. LuxR, the high cell density one, is turned off when phosphate is present. And when autoinducers bind and drain phosphate, LuxR turns on, AFA turns off. There are some other secondary regulatory components, but for simplicity's sake.
Cool. Thank you.
And then just to answer Elmar's question a little bit, at least in our model, the mutation could be from any cell type to any other cell type. And that was based on the Sauron model that we read. So similar to how they did it. So we didn't have any restrictions for the kinds, but I'm sure that's biologically validated.
You kind of pointed in that direction at the end, but I did feel at some points that maybe you had too few cells. So my question is, how long does it take your simulation to run to completion?
That depends on what you define as completion. It runs fairly quickly, at least on my operating system. So like if I'm doing like 10,000 MCS, that will run in like 20 minutes, depending on like what I'm doing. So I can run quite a few in a day, which is nice. Yeah, that's if you have like another question about that.
Yeah, no, so 20 minutes is still pretty fast. I feel like you could make your simulation 200 by 200 without too much pain, probably.
Yeah, I've tested it with larger lattices, just for like in terms of like quickly tuning those parameters. We kept it small to keep it running quickly. I tested it with 500 by 500 lattice, just for my PI wanted to see some things. And I was able to run that one at 10. I think it took like 45 minutes to run the 10,000. I can't remember exactly, but it was at least double the time, but it was still less than an hour.
Okay, thank you very much. That was great. We'll move on to Gabriel next, I think. You ready?
Yeah, so I don't know how I'm supposed to follow that one, but I'll try. Here we go. So I'm about to say something I'm sure that will send a shiver up James's spine, and that is so are you guys able to see just the presentation, or are you seeing the like presenter window?
Presenter mode.
Presenter mode. All right. I'm using Connor's here actually, but does anybody know how to not launch it in presenter mode?
If you do launch it, I think there may be some options.
So launch it just as slideshow, and now go reshare, and you should have an option to just share the
Other
power-
side.
Gotcha. Let's. All right, how does that look? That's good. So I'm gonna say something that's gonna probably scare James, and that I started this over probably like last week, right? Because or like actually no, not even last week. I think it was this weekend. It was only a couple of days ago. I started from scratch because so just a brief recap, I'm doing optimization within CC3D. The model that I was presenting to you last time, while it did work, it would have taken forever. Like it would have taken, it would have taken a very long time, and I don't think it would be very fun for a user to look at it as an application. I don't think it would be very enjoyable because it would be doing nothing probably for a vast majority of the time. So I basically started from not from ground zero, but pretty close. So also a huge thank you to Pedro Castel, because I built most of this off of his chemokinesis code, so his run and tumble simulation. I had one, but I decided to go with one that was a little bit more, I like the way that he did a couple other things, so just saying that right off the bat. But yeah, so here, we'll walk you through. Man, presenter, so I am not used to PowerPoint at all. So basically, fundamentally, this project has been about how can we put optimization within Computational 3D? So how can we is there some sort of neat, reproducible, practical way to be able to put optimization methods, whether it be not evolution, but differential programming, machine learning, deep learning, how can we put that within CC3D and kind of have it as like, almost imagining something like the little boilerplate code snippets that you're clicking on the side. Is there a way to do that with optimization, right? A little bit of a motivation for why optimization, even though you guys have heard me say this last time, we can parse through large data sets, our simulations are getting more and more complex as time goes on. So does the data, right? So, and in many ways, we can, it helps us look for things that we might not be able to find just off of the naked eye, right? That's the kind of motivation that I like to give. We'll come back to here in a little bit, but the way that my traditional spiel is that I think of this thing as the infinite parameter space, right? Biology gives us some sort of subspace of relevant parameters, and then optimization gives us a little bit of sets within that subspace to look at more thoroughly as they're related to a task more specifically. So that's just a basic idea. Why do we want to do this? This is kind of the idea, right? But these things get narrowed down the more that they're reflected in the actual behavior of what you're trying to do. So this is why we want to try and keep it within, right? Within CompuCell 3D, rather than train something outside of it, then put it in. It's most likely not going to do what it was doing outside of it, right? So again, establishing that base case, decided to go with the run and tumble simulation because it was a straightforward way, wasn't going to be too computationally expensive. But this, again, the goal of this isn't really just for, putting a run and tumble simulation in CC3D, but it's, okay, can we establish some way of putting optimization, deep learning, something like that within CC3D for whatever, I was actually just reading about, trying to find optimal tissue patterns for bioprinting or tissue engineering and all these kind of stuff. Optimization methods could probably help with that, right? We've seen some papers that do that already. So I'll just do a very quick walkthrough of the run and tumble simulation from Pedro. But basically, what we're giving the algorithm, is 3 parameters. We're going to be, we're only giving it one thing. We're giving it, it's what the concentration is of the field at its location, and it only gets a small memory, only 10 time steps, right? And just the change in the field concentration at that time, or some sort of change in the field, right? So we can then put that, and so we give our bacteria 3 different things. So it can as output, it can do persistence, which is basically related to how much the cell will change direction as it tumbles. TP, which is the actual probability of the cell tumbling at a given point in time, and P itself, which is just the polarization, the kind of vector, the angle of direction that the cell actually goes, right? So these are how we're mathematically defining them. So basically, at any given time, the cell is asked, if some random uniform distribution in between 0 and 1 is less than the probability of you tumbling, then you'll end up tumbling, right? And when that happens, you basically an angle is drawn from a distribution, and then you're getting shoved because of that angle, right? You're getting offset on that angle. So just to show you what this ends up looking like when we end up putting this in a simulation, is you end up seeing a kind of pseudo-random, not actually random, but it ends up going towards the concentration over time, right? So it'll run to the right over and over, and because the edges are periodic, it'll actually loop over. This is something more of like a torus rather than just a 2D grid. So yeah, so this is what a kind of basic run and tumble simulation should end up looking like. So given this, the question then becomes like, okay, what would we need to give? So we need to give the artificial intelligence the input, which is simply just that field history and the change, and we have to decide what we want it to choose to do, right? What kind of outputs do we want it to have? So this is when we go to make it. So what we're doing is we're using TensorFlow, which is a very, it's all in Python. Its backend is in C++, so it's fast, which is very nice. And before I was doing this a very different way, where I was using more of a machine learning approach, trying to reduce the error between what I saw was the angle or the angle between the cell and what the correct angle would be. But rightfully so, this isn't, in an effort to make it more biologically realistic, even though this is, more on the edge, on the boundary here, but in an effort to make it more interesting, it's much more interesting if we actually just if we let it play with those things. So if it simply knows its field history, like in the previous simulation, and it has to balance something between the vector, the probability it tumbles, and its persistence. It has to find some sort of balance in order to do a good behavior. What does it end up doing? So also, what I should say is that I switched what approach I'm doing. So this is what I spent my weekend doing. Was basically I was implementing a reinforcement approach. So this is a very popular approach for a lot of stuff. Like they train Mario to play, or they train an AI to play Mario. They do all this different stuff, right? It's usually reinforcement learning. So, what is this, right? What is this computer science math that I'm throwing at you? Basically, all you really need to know is that this is an extremely simple, a bare bones version of a reinforcement learning algorithm, because I didn't want to get too crazy and have it do, ridiculous stuff that a bacteria would never do. I wanted to keep its memory very short, so it still had the same thing as you saw in the previous simulation. It still has a memory of 10. And we basically kind of define its state, action, and reward as these three things. So its state is its field history and field change. Its action is the self.model.predict of the state, which is basically what that's doing is it's taking the state as an input and returning a list of Q values, which is then, the argmax is done on that, and then it's chosen as a action to perform, right? And then after that, the reward is calculated. So the reward is taken to be the field value divided by the max field value, and then subtracted by some sort of penalty term. I just for information, I always defined the penalty weight as being 0.5 because I didn't want to get too crazy. So basically, what that means is that if it goes in the no concentration area of the gradient, it gets a little slap on the wrist, right? It's not getting destroyed, it's not getting exploded, but because if we drove that penalty weight up, it's not just going to get a slap on the wrist, it's going to get smacked out of existence, right? So if we need to balance some sort of like, okay, what's the right little punishment? It's not that nice. So it's gotta figure it out using these things. So what I'll do really quickly is I'll just pull it up over here. All right. So I just want to briefly explain some of the plots that I have here. So this is the loss over time. It has a pretty basic definition in reinforcement learning, which it's just the target. So where we want it to be. So a kind of good action to take, subtracted by the current Q value. So what it's doing. So basically, are its actions getting better? Right? Is it choosing kind of the right, is it making like a right decision, right? Here's the reward over time. So this is you're gonna see that actually, if we let this play a little bit more, it's not, there you go, okay. It goes below 0, and this is because of the penalty term. So this is because it can get punished for what it's doing. So which we can see as it dips there on the right, it's because the cell is going over into the blue side. And then we have a plot over here that shows the cell position, just for kind of like a sanity check. Where is the cell? Is it spending more time over here? Is it spending more time over there? So we'll just let this run for a little bit. So at the beginning of the simulation run, the vector, the probability of tumbling, and all of the parameters in the model are randomized. So they're randomized between a min and a max value, and basically what the network has to do is figure out, okay, I need to change these parameters in a way so that I can maximize my reward and I can decrease the loss over time, right? How can I do that, right? So, and it's only given those three things. And I'll show you a little snippet in the code that I think helps understand this in a second. Or actually, I'll show it to you right now while we're waiting for this guy to give us a little bit more data. But it trains, this one I was actually very happy with. It was a huge relief to me over this weekend because you can see it immediately starts to perform better. The loss shows over time, it's going down, it's actually training. Wow, there's this great sanity check, it's learning, right? And what's even more surprising is it does in a very reasonable amount of time. It's only been, sorry, the thing is blocking it, but it's only been 5,000 Monte Carlo steps, and it's actually improving, right? Just super interesting. It was super exciting to me that I actually got it to work, because there was a little bit of a panic over the weekend when the time was going crazy, and I was just like, okay, we're all right. We live another day. But yeah, so here I'll show you a little bit of the snippet from the code for the actions. All right, are you guys able to see the code? All right, cool. So basically this function that I put right here called update cell properties is basically, so the output of the network is any one of these 6 actions. And what that could be is either increasing the P, decreasing the P, which is the polarization, so this is the direction, increasing the persistence or decreasing the persistence, increasing the probability of tumbling or decreasing the probability of tumbling. And then I just have these functions here that just make sure that it doesn't get to out of bounds. It doesn't either, it doesn't do something that's, quote unquote illegal, right? So I wrap an angle and then I clamp it so that persistence doesn't go somewhere crazy. Yeah, so then we can go up here. So this is set to 20 MCS right now so that it runs in a reasonable amount of time. But this is exactly the function that very computer sciencey function that I showed you before is happening exactly here. We can see that. We have the state, which is defined as the field history and field change. We have an action, which is the self.predict of the state, and then we argmax that. We update our cell properties, so that means basically the cell makes a decision. It does that. We then evaluate its reward. We add a data point. We calculate, we add some sort of a memory that's for training purposes. It's not actually, this isn't the cell's memory. This is more for like batch training. Bad wording there. Should more be something else. And then we update the model, which I'll show you this function really quick. Here's the compute reward. And updating the model, and this is where the actual training is happening. So this is where the 2 different types of Q values are being figured out. Yeah, so basically this right here is similar to that function that I showed you in the presentation. Got an action, gets a reward. Gamma is simply, it's also called the discount value. So it's basically, how much is the value for this action? Yeah. Oops. So let's see how our guy's doing. Reasonable. So it's actually pretty cool and what's fun. So again here, the cell has a very limited memory. So the fact that it's able to, I was impressed that it was actually doing as well as it's doing. So it doesn't go crazy, because so something to note is that the force will not change, because if I give it the force, all it's going to do is walk over to the highest point in the gradient and stop moving. That's it. It's very boring. So I just put the force as constant 200. It has to figure out some different ones. And what I love about it too, and this is why I love this kind of stuff, is is time you initialize this, it's going to have a different behavior. So there's some that they start and they just think that the best strategy is immediately just to start spinning. And they're just doing donuts the whole time. And they just and they actually perform pretty well because, another stable strategy is, again, what we saw in our probabilistic simulation, the original run and tumble simulation, where you saw the bacteria just continually swimming over to the right. That's a very stable strategy. We can think about these different strategies are basically, when we think back to this graph over here. You guys able to see this? Is, a lot of it. So when we see it running to the right or doing some other thing, we can imagine that as one of the smaller hills. It's a local maximum, right? Because we can sit here as an observers and look at this and say, okay, why isn't this cell, obviously there's some sort of global maximum here, which is where if the cell gets up here, if it continues to run up, it will always stay in the red, right? It will always stay in that continuous value. But that's a really, really difficult thing for the cell to learn to do, because what is it using? It's using its field value and the change in field over time. So to do that, a very stable strategy is just to run to the right, because it knows that its field's changing over time, and it knows, it knows it can actually predict that. It's hard to predict when the value is constantly the same, what's happening, because nothing is technically changing from that point of view. But yeah, just super, super interesting.
Not to interrupt you, but I think that it kind of learned to just go up and down.
Oh, did it really? That'd be awesome. I haven't seen one that's done that yet.
I mean, it still goes a bit left and right, but it's mostly going up and down.
Yeah, we lost him. Or is he coming back? Yeah, believe me, the amount of it's so easy to just stare at this for hours and be like, my son is learning. He's doing it. And then it's and then you're like, oh my God, I need to finish this project. But yeah, so I have a couple of models that are saved, but the only thing is that they pretty much do. I have one that is 500 MCF or 500,000, sorry. That's been trained for 500,000, but it does practically the same thing here. What we see is that what I'm finding is that, and this is pretty much because, and I have a theory, is because the reinforcement learning algorithm that I'm using is so simple, it's so bare bones, that a lot of this stuff. So something that you would do if you really wanted to get it to find the global maximum here is you would start to experiment. You would start to say, okay, we need the cell to have a larger memory, we need to do all that other stuff, or we need to, there's another strategy. So this is also, to use lingo, this is an epsilon greedy strategy. So basically the cell is only going to do what's immediately satisfying to it, because it has no balance between exploration and exploitation. So this is all, more cognitive science lingo, but all that means is basically that if we want the cell to get out of that little hill that it's falling in, and we want it to try and climb up a different one, a much taller one, we're going to have to start thinking about different ways of pushing it out of its comfort zone, basically. Yeah, it's very fun. All right. So that's what I have for today. I would show you the NanoHub application, but I'm in the process of bouncing back and forth with what is it called? What are they? Tickets? Because I'm trying to get it actually published, which Juliano, I was probably going to hope to talk to you maybe a little bit about. But yeah, I would love to answer some questions or talk through any more of this. I figured I'd leave it more open framework. So then, but yeah, it's just fun. I just had so much fun with, when I was actually seeing it do different stuff. I mean, I see many of the applications that I see to this is, again, because, in so few lines of code, and while also being able to do it in a reasonable time without being so computationally expensive, There's many different stuff that's much more biologically meaningful than this, right? There's many different things that could be very, very interesting and related to experimental work that I'm really trying to establish tight theory experiment loops between, now that this is, this proof of principle has been shown. But yeah, anyway, I just got extremely excited when it was doing this thing and when I was like, finally, it worked. But yeah. Oh, I'm sorry, I missed all of these messages. There's 19.
Fake you took care of it.
Okay.
So I have a question that might be just like a lack of cognitive science understanding, but can you, or TensorFlow understanding, can you set it so that like it will, like I think maybe it was Juliana that talked about it, like different forces, so like it'll stay and then like move around, but can you like, yeah, I guess like my question is like, especially like when you think about a simulation with multiple cells, right, like breaking off from like a clump at high nutrient levels to like explore new niches, like have you thought about how these simple lines of code begin to like have emergent properties when you add in more cells, or like how, yeah, like you were the biologically relevant phenotypes you were talking about?
Yeah. I mean, that would be really fun, right? Especially because, so I mean, so the reason why I tried to stick to one network is because this is one network, and the more networks you add, the more computational expense it adds as well. Because this is technically this isn't just Q learning as in reinforcement learning. This is deep Q learning, but kind of. It's a to be specific, it's a it takes an input of the form of the field history, and then there's 64 nodes connected to 32 nodes connected to those output actions that you saw. So it is the deep learning approach. So it will, if I had multiple of these, I'm sure that it would take much longer than what we've seen. But no, for sure, I 100% am super interested in those types of emergent properties, social interactions. My research actually is on evolving minimal robots to do stuff. So that's definitely very up much my wheelhouse. So I'd love to talk about it more if you're interested. It's just cool.
Other questions? I think that's great. I was saying that at some point it would be nice to have the source move slow, have a point source that has a long diffusion, like, and have it move slowly, so it's not always having the same environment. Having the cells consume the nutrients, so it has to keep moving in order to be able to survive. Sort of combining what you and Logan were doing.
So Joel and I actually have code for, so I haven't tried it with this new version, but for the previous version, we have code for basically whenever the bacteria would reach the food source, it would disappear and then spawn somewhere else randomly. So which was really cool, because when you trained the previous one after a very, very long time, you would start to see that it would start to do things like it was really neat. It would start, because it knew that it was on a torus, it would start to, as an observer, you're like, what's it doing? But then out of nowhere, you're like, oh my, like it just found some angle that I didn't even imagine. It's just pretty neat. But no, definitely that could be added pretty simply. It would be cool. I'd be interested to see how it would do.
I think your simple neural network one makes sense. It's a bacteria. It shouldn't be smart. Two, I think even that should be enough for it to learn the moving source and learn to turn its speed down when it reaches the source, because it's going to be a high concentration. So if concentration is high enough, I'm just going to stop. And
I could
later.
add, if you guys are interested, or if anybody's interested, I can show you what it looks like if you give it control of the force, because it is funny. It typically, it will just run up to it and then stop immediately. Well, it does some sort of exploration at first because it doesn't know what the maximum is, but typically then it'll do the thing where it'll just run up to it and stop.
Right. Well, that's why having the food source move is interesting.
Yeah.
Because in that case, just finding food and then stopping forever isn't the strategy.
Mhm.
Right?
Yeah.
And some bacteria are very
Works for plants.
complicated. Well, that's true. Plants have different strategies, but I mean, some bacteria they'll build motile. They have sessile and motile phases. When the environment is good, they stop and they reproduce. When the environment's bad, they put their energy into moving.
Yep.
So they actually do this. And sometimes, depending on the species of bacteria, some of them have very elaborate motile machinery, the type 4 pili and things like this, where it takes them a lot of energy to move. And they'll assemble all of that when they're in bad shape and they need to find food. When they find something good, they'll disassemble it because it's expensive, just the way Logan was talking about. Producing the public good is very expensive. You don't want to do it if you don't have to, but it does allow you to use resources that you wouldn't be able to do otherwise. I was thinking the other day, Dale Kaiser, in the old days, did a lot of research, experimental research on this topic in the context of mixing bacteria. And actually here at IU, Mike Lynch did some things on this, and he left for Arizona State, and Clay Fuqua and Yves Brun all worked on some of these problems, and of course Logan's advisor as well. And of course your cell could represent an animal rather than a bacterium, so it could have additional, could be smarter, it could have additional intelligence perhaps. Although bacteria are basically good at integrating multiple sources of information. They may only have very few output channels, those couple of parameters, but they integrate. If it's toxic, what if the food source is near something that's toxic? What do you do? These kinds of things. So I think, as you say, as a proof of principle, this is great. I can see extending it in all sorts of neat ways. The next, we have the Zebrafish team. Looking forward to seeing how that's developed. And I'll turn the share over to you.
Okay, so we are Team Zebrafish. And for our project, we chose to look into modeling zebrafish stripes using a couple different models. So for this presentation, we will go over our learning objectives for the project. We will talk about some background biology, talk about simulation backgrounds, and then we'll sort of do a comparison of our initial model versus the newer implementation, and then do a code demo, and we'll go over our educational exercises. So for our learning objectives, what we want to do is to understand how the three main pigment cells of zebrafish stripe formation grow and interact to form these stripes and their patterns. So for that, we want to know the biology behind stripe formation. And to do that, to model it, we need to know the mathematics behind modeling such stripe formation and how any chemical field interactions come into play. And then we also want to know how to test for stripe formations, adjust those interactions and parameters to make different patterns. So the paper that our latest implementation is based on is modeling stripe formation in zebrafish, which is an agent-based approach. This work was written by Alexandria Volkening and Bjorn Sandsteed. And before we get more into that, we will go into the biological background. So you may remember from last time, there are three different types of cells that govern stripe colors in zebrafish. Those are melanophores, which correspond to black color, xanthophores, which correspond to yellow, and silver iridiophores, which sort of help guide the formation of the stripes. But they're also based on cell-to-cell interactions and the range between cells. So long-range interactions occur in adjacent stripes and in yellow stripes. And like I said, the alignments are aided by the iridophores. And also these stripes seem to grow and widen and change as the fish develop and age. And we're also looking at how repulsion between melanophores and xanthophores maintains black and yellow stripes separation in their boundaries. And so this is some background on one simulation that was tried to address this problem. The melanophores will release these short-range signals, and those inhibit the growth of xanthophores and mildly inhibits growth of other melanophores. Xanthophores will release short-range signals. And these also inhibit the production of melanophores and mildly inhibit xanthophores. But xanthophores also will release a long-range signal, and that is actually required for melanophore survival.
So here we have mathematics behind our previous simulation. So you can see on the right, it's like the code that we have in our XML. We actually aren't using this anymore. We still left it in there because it is one way that can be done. So I'm gonna just go over a little bit again, just if people like do want to use the simulation, they can. So in here you can see that it's modeled off of a reaction diffusion with 3 different factors. With U and V representing the density of melanophores and xanthophores and short-range ligands, and then W representing the long-range factor. That's definitely like the most important part about it, and that it does the minimum of the maximum representing piecewise functions. So that's pretty much all I wanted to touch on based on that, since we did go over it last time, and we'll go over it a little bit later, but we're actually not using this part of the simulation anymore. So here's some basic background code information. So this is still the same. We still have 128 by 128 doing 10,000 steps. We still have medium secreting cells, non-secreting cells, melanophores, xanthophores, a wall, and then also melanophores 2 and xanthophores 2. Last time they were a more like mobile version of the cells, and we did switch up a little bit the meaning of them, which we'll go over later. And then here are our contact energies. So this still stayed the same. We still have the same contact energies as last time, and certain ones are increased so that cells can separate. And the contact energies between, sorry, I've been a little bit sick. The contact energies between the same cells are greater than the medium, so they'll separate more. And then here is a little bit more about our initial simulation. So we did initialization of chemical fields, and we seeded cells based on chemical field thresholds. So here are like previous results from educational exercises. And so you can see like the different fields. There's U and then V and then W. So this is the section that we like kind of switched it up on. And then here's just like a little demonstration of our previous results. So you can see here, there is cells seated in the cell field, and each of the fields are being made. And then as we go through, more cells develop and start to form that stripe formation. But it's mostly based off of the different fields, not as much what the cells are doing themselves.
Yeah, so I guess after having created our previous simulation, it wasn't as impressive as we initially wanted it to be. So what we really wanted to do was try and recreate the UVW type field behavior, exclusively looking at like cell-to-cell interactions. And when we were looking at the paper by Alexandra, she had done something similar by giving each cell type 2 cell fields that correspond with them. A long-range a long-range chemical field, and then a short-range chemical field for both melanophores and xanthophores. And the short-range chemical field basically has a diffusion length that is a third, or approximately a third, that the length of the long-range one. And then by using the interactions between both short range and long range of the melanophores and xanthophores, you can kind of create these different parameters that can generate similar behavior to the UVW type simulation. So some of the changes that we did is we are no longer seeding cells into the UVW field. In fact, the UVW field basically don't exist in our simulation anymore. Instead, we are seeding them based on interactions between the short and long range melanophores and xanthophores field. And here you can see just some of the names. We have xanthophores and melanophores 2. These are the short range interactions. Xanthophores and melanophores 1, I'll call them, are the long range interactions. And then our cell death is also no longer related to UVW fields. It is now directly based on cell to cell interactions. And then our thresholds as well are based on now these new field types that are exclusively governed by the melanophores and xanthophore cell types. Okay, just taking a look at the XML briefly here. It's about the same as we had mentioned earlier. I'll go in our code demonstration, I'll go more in depth into this. Basically, what we need to note that's important is this in the uniform initializer, we basically just have our initial conditions. And this is kind of something I think Mike will talk about this later in the educational exercises. This is probably the most important part of our XML, which is initializing your conditions to see what patterns form as a consequence. And then here we can also just see, this is the melanophore 1 field, which has a diffusion constant of 1, and our, well, you can't see it, but we have 3 other fields. And the only difference between melanophore 1 and melanophore 2 is the diffusion constant is a different diffusion constant, and that helps us get the difference in diffusion length, right? And then xanthophore 1 and xanthophore 2. Xanthophore 1 is exactly the same as this, basically. And then xanthophore 2 is just the shorter range version of xanthophore. The steppables, I'll go more in detail into this when I do the code demonstration, but basically, sorry about that. We are just looking at, these are just different thresholds that we care about. And then basically there are, I guess, 3 important parts to our steppables. One of them is cell death checks, where we check to see if the cell is no longer in its appropriate environment, and then we will give that cell a certain amount of time to move to its appropriate cell locate or appropriate field location. And if it is no longer in that location after that time check, then we'll kill off the cell. If it is in the right location, then we leave it. The second important part is cell seeding, which is basically just we are searching through the field for empty locations, and then there's a certain probability that's based on our seeding rate or our creation rate that will will fill the field until right now it's 95% of the cell field is full of stripe or full of cells. And then our third important part is just the seeding parameters. And this part is probably the most important part of the whole code. Basically, we are just analyzing, these are just the new parameters that we had to implement that are exclusively based on these new 4 chemical fields and their interactions, and how their interactions can give us the type of behavior that we were getting from the UVW type simulation, but instead we are getting it exclusively from these new field interactions. So I'll unshare. I don't actually even know if I need to unshare, but I will. Can you guys still see my screen? And you can see the twit. Okay. So I recognize that the cell seeding is at the end of our steppables, but it's probably the most important thing to start with. Basically, the way our new cell seeding works is we are no longer looking at concentrations of UVW. Instead, we are looking at kind of distance and interactions between other melanophores and xanthophores, which is the information that we end up getting from these new short-range melanophores and long-range melanophore cell fields. So the first thing that we need to check is there are two different conditions that will yield in the generation of melanophores. So this is the generation of melanophores. Basically, if the short range melanophores is greater than the long range melanophores, then you generate melanophores. And this basically just means that if you are close enough to other melanophores. So this is basically saying melanophores like to spawn close to other melanophores. And then the second condition, so this, if you just had this, basically you would just start generating a melanophore stripe, and that stripe would kind of just persist. You would just have like a stripe of melanophores and nothing else would happen. And melanophores would only generate if they were close to other melanophores. So now we have this second condition, which also says basically melanophores will also generate if they are sufficiently far from other xanthophores. So this is the opposite kind of condition, where basically we were saying that the short range xanthophores is less than the long range xanthophores, which basically means that we are far enough away from xanthophores. And then we also have just this extra threshold condition. Basically, that's just to say far enough away. So basically, melanophores spawn close to other melanophores or far enough away from xanthophores. And then here, our xanthophore creation is basically just the opposite. It's just xanthophores spawn close to other xanthophores. Oops. Yeah, basically, xanthophores spawn close to other xanthophores, or they spawn far enough away from other melanophores. So that's basically how our cell seeding works at the moment. And then our cell death basically functions similarly, but we are basically just checking to see if, well, it might be a little confusing because I'm no longer using the same variable names, but basically we are generating a random seed. We are checking the melanophore concentration at that little random seed, and we're checking basically the concentration of all four fields at that random seed. And if at that random seed, all the concentrations meet one of the two requirements, we are spawning a cell there. And then if a cell already exists, we are basically checking the field concentrations at the center of mass of that cell to check to see if it meets the conditions for like cell type change or cell death. So basically, if a melanophore is super close to other xanthophores, we are switching its cell type to this other cell, which we're calling melanophores 2, and melanophores 2 chemotax towards melanophores cell field. I recognize that might be confusing given that they have the same name, melanophores, and melanophores 2 is the same as their chemical field concentration names, but they are different. Basically, melanophores will become chemotaxing versions of melanophores if they're close to xanthophores, and then vice versa for the xanthophores, they will become chemotaxing versions of xanthophores if they are deemed to be too close to melanophores. And then here, basically, we are giving every cell 300 Monte-Carlo steps to relocate themselves to appropriate locations. And if they do relocate themselves to proper locations, they switch back to the quote unquote stationary, not actually stationary, but non-chemotaxing versions of their cell type. And if they are still in the wrong location, we kill them. So ideally, I recognize there is a bit of a non-biologically accurate part of this, which is that we are just doing it every 300 Monte-Carlo steps, which means that if a cell change type at 299 MCS, they are given exactly 1 MCS to move to the right location. So I was working on this earlier, basically just having a cell dictionary for any of these cell types. When they transition, we would basically be counting up their individual cell counter, but that slowed down the process quite a bit. So for the sake of speed, because we do have a lot of cells in these simulations, I guess made a little, we just cheated a little bit and just made it kill off at 300 MCS. So this is more problematic when there are few cells in the simulation, because I think when we do the cell demonstration, Mike might show this, but at low mod 300 MCSs, you kind of get more cell death, because I guess the cell field changes are more rapid, but kind of as you hit like 3,000, 4,000 MCS, you stop getting large cell death because the fields are kind of equilibrated at that point. That's basically it for the code. Next, Mike is going to talk about some of our results.
All right. Yeah. So so here are the new updated results. So you can see this is the outputs. The top middle is a cell field, so that's the actual cells. That's the main output that we're looking at. And then we have those 4 different chemical fields, the short and long range chemical fields for both the xanthophores and melanophores. And so yeah, so this frame right here is like 23 steps in, so right after you started off, and you can see that straight horizontal line of melanophores, and then you can see in the melanophore chemical fields, you can see those secretions, the long range and shorter range fields, and then nothing in the xanthophores yet because there's none seeded in yet. But as we go along in the simulation, you can start seeing the cells seeding into the environment, and then you can see those fields start to progress over time. So melanophores, and then you can start to see those xanthophores. Yeah. All right, and then as we keep going, you just kind of see more of that happening. You can start to see the stripes forming in the cell fields there, and you can also see the chemical fields, and how it's still a little difficult to tell now, but you can kind of see the where it's high concentration of melanophores, it's low concentration of xanthophores, and yeah, and I think there's a few more. This is like at just over 1,000. Now this is getting to 2,000 steps, and you can really start to see those stripes forming in the cell field. You can also see the green cells. Those are the transitioning chemotaxing cells there. So right along the edges of those fields. And yeah, and now you can start to really see the difference in the xanthophores and melanophore chemical field secretions. And I think we have one more step. Yep. And so this is kind of nearing the end, when it's almost not fully seeded in, but this is where we have most of the cells in the environment. And we can really see those nice stripe formations in the chemical fields now. And you can see that inverse relationship between the 2 chemical types, and how that shows up in the cell field.
Sorry, just briefly, I just want to mention that one of the limitations that we are running into in the simulation is you might notice that you are kind of seeing some stray xanthophores at the edges of the stripes. And this is, I suspect at least, attributed to the fact that the domain that we are running this under is not generating the entirety of the second stripes, because so there is never a great enough concentrations of melanophores to fully expel the xanthophores. So I ran the simulation at a larger Y value, so like 200 high, and then you get the full second stripe formation perfectly fine. You don't get the intermittent xanthophore. So this is just a byproduct of the limited cell domain, or the cell space or field space, basically. You can go back, Mike.
Yeah. All And yeah, I think we can see that more clearly in the next couple slides. Yeah, like just looking at the cell field. So yeah, so this is just the initial startup. We have that line of initial cells, and then they start to kind of move around a little bit, and then yeah, and now we can see the xanthophores start to be seeded in. We can see a bit of changing of the cell types there, and then as we keep going, you just see more of that happening. You can also see in these close-ups the wall around the edge there. And that's also another part that kind of, limits the size of the simulation. And if you recall from our initial outputs the first time, the stripes were a lot more, they were a lot thinner. And I mean, those stripes were just, us manually putting in the chemical fields based on the equations. And so they were a lot more, I guess, like more precise, but less like biologically accurate. But now that they are based off of the cell interactions, they're a lot more biologically realistic. And so that's kind of why we see they're larger stripes. And this reflects the paper too, because actual zebrafish stripes, I can't remember exactly the number, but it was like what 7 to 12 cells in width or something like that. And so that's pretty much what we're seeing with these updated relationships, that as the cells secrete and as they move around, depending on the short range and long range chemical fields, that's the size of the stripes that we see showing up. And yeah, of course, with the initial simulation, we could have like manually made those stripes larger, since we were just manually putting in those fields anyway. But this kind of shows that this is the actual biological representation. And we can also change the width in this simulation as well, which I'll kind of talk about later. Actually, I think right now, yeah. All right. So I'll go ahead and share the NanoHub. All right. All right, can you all see this fine? All right, cool. So yeah, so I had this running kind of in the background a little bit. It's only at like 2000 steps in, but this is kind of what it looks like when it is, pretty much simulated out. So we have the cell fields here, and then the melanophores, and then we can switch and look at the xanthophores. Yeah, and then I'll go ahead and start it off from the beginning. So this is our NanoHub page here. Can launch it, and we can maybe see if we can see that effect at 300 steps in. Let me just make this larger. All right, so yeah, we still have the UVW fields, but if we're just looking at, hold on, yeah, make this a little bit easier to see. It's running kind of slow on my laptop here. All right. So yeah, so we have the short range and the long range, and then the cells in the middle there. And I don't know how fast this is going to run. It's okay. It's good in there. So yes, we see the melanophores there, and then we can change. So I had a like a steering panel here, but we had different versions of the code uploading, and this one did not have that updated one, so I'll have to re-upload that to NanoHub soon. So here we're coming up on 300, and we just see the melanophore cells there. Eventually, we'll start to see the xanthophores seeded in. But we can look at the diffusion fields here. So we have the 4 different types, the short range and long range for the melanophores and xanthophores. And so we can go ahead and change those in here. So the 1 and then the 0.3, so the third of the value for the ratio of short and long range. And I guess I'll just kind of like show what it's like to change that. I'll just make it like decrease it by a lot, and you should see it yeah, start to like drastically go down. That might really screw up the simulation, but yeah, you can see a proof of concept there. And yeah, now we see the some xanthophores coming in there. And so we should be able to see some of the chemical fields there coming out. And yeah, it's running pretty slow, so it'll be a while until we actually start to see the stripes coming out. But yeah, so I think that's good for the NanoHub side. Let me see. I can go back and talk about so some of the exercises that we had. Yep. So similar to what I was talking about last time, we had the initial starting regions to create the different stripes, different patterns. And so that is done in the XML. We have the different starting regions of initial cells. This is the example given in the previous simulation, but it's the same principle in this version. Because it is of a larger scale, the patterns are harder to see, but in principle, it is the same effect. So yeah, so this is just exploring, creating different shapes, seeing what you can do with that. Also, another big part is, as I mentioned before, changing the actual characteristics of the stripes. So like the width, and yeah, so by changing the parameters for the long range and the short range chemical fields, you can create the different structures of the stripes themselves. So you can kind of see in these examples, it's a little hard to tell with these, but the thinner stripes of melanophores there, and then larger in the xanthophores, and vice versa. All right, yeah. And so this is kind of the same thing, but building off of that is another exercise is to just observe the change in chemical fields, which, yep, there we go, over time. So to kind of see it as the development of the zebrafish from a young zebrafish to an adult, and how the stripes actually develop biologically over time. And as you can change the parameters to kind of see the different effects that it has and how they solidify over time, and the relationship between the chemical fields of the melanophores and the xanthophores, and how they're kind of inversely showing up in the fields. All right, and then I was going to share the let me see how to do that. Just a little bit of the document that I had with the exercises here. So I know I kind of mentioned this last time, but these are kind of the different regions that we have to initially start off the simulation, and how they can create the different shapes. So those are added in the worksheet for the exercises, just as a template, and kind of how to create your own kind of regions to explore the different shapes. And then just some suggestions for parameters for different.
I don't think we're seeing your worksheet, Nick.
Oh, are you not seeing the Word doc? Oh, okay.
Yeah.
There we go. Thank you. There we go. So yeah, so this is what I was talking about. The different shapes that could be created. And then I just have like examples of the different parameters that can create the different effects down here. So yeah. And then I'll go back to the presentation. And I think that was about all we had. Yeah.
Yeah, maybe just before we end, I can briefly talk about, I guess, areas of improvement for the project. One of the major areas of improvement that we would really like to do is try and run this on bigger simulations, because as you can see, I guess, from some of our actual images, if we have cells that are 7 to 10, or if we have stripes that are 7 to 10 cells thick, I think 7 to 12, I think ours are like 10 or 11. But if you have that, if you want stripes that have that many cell thickness, you tend to only get 2.5, 3.5 stripes formation, which in this 128 by 128 field, which is a bit limiting in terms of how much patterning you get to see. Because if you remember our just UVW field simulations, we had like, I don't know, 11 stripes, something like that. And it's way more interesting to see pattern development this far out in comparison to just 3. If you like zoom in on this to like 3 stripes, I mean, and we initialized the same patterns that we had on like the left graphic. I mean, you might see like 2 dots, 3 dots, which is really, I guess, not very insightful. So I was communicating with Giuliano a little bit, trying to figure out how to optimize our code to really be able to utilize a greater field space. So it's, I guess, the problem is we are just seeding in so many cells into the simulation. Like, the nature of the stripes is that they want cells basically everywhere. And that is not nice for CompuCell when you have thousands of cells in. So I guess there's a couple things we could do is we could like just run it on, PhysicCell can handle more cells. Yeah, maybe we can try to port it to PhysicCell. That's one possibility. But also maybe just run it on like Big Red, something that has more computational power. And then we could really see these like larger patterns, which I think would be useful. That's, I guess, the main next step we kind of want to do, which is just being able to visualize patterns in the larger scale. So we tried to multi-thread the code, but Giuliano, I believe, said that NanoHub only has 2 cores, which is not faster, I guess, than not multi-threading the code. Oh, 4 cores, okay. But I guess it didn't give us a significant increase in performance, at least locally, or at least that's what I noticed, at least. But I mean, I definitely think if there are ways to improve the code, I would definitely like to continue doing that, because I personally want to see how this can like develop, because it looks quite nice, I guess. And I would like to see it look nicer at a larger scale. But yeah, any questions, we're open for them now.
I think you laid out possible next steps really well. It's impressive how much progress you made in the past 2 weeks. It's really, as I say, I'm sure Alexandria would be very happy to see this and probably have some neat ideas about what could be done with it. So, and you know, when things settle down, I could maybe Giuliano and I could look through the code and see if there's any way to optimize things a little bit. Because you don't have that many cells and it's 2D, so it shouldn't be that slow.
I think the issue is the chemical fields.
Because they have long diffusion lengths.
Yeah, and the reaction diffusion, I guess, also is more costly.
Right, but you can turn that off in the so I think, I mean, it's not gonna make an enormous difference, but you could probably speed it up. A factor of 2 probably, not that if we were careful.
So if you want to look into porting it to PhysiCell, I could help you. I mean, I know in the meanwhile this software quite as well, but if this is as well to compare what it can do, how it looks in PhysiCell and how it looks in CCD might be quite interesting if statistical mechanics has here really some say, or if you just can modulate it with the classical mechanic. I think that would be interesting. Yeah.
Yeah, I agree. I think maybe porting to PhysiCell might be a good idea if we can do these larger scale things better. I think that's a good idea, Elmar.
I have a quick question.
I'm
out
happy
of
to help and
curiosity.
sit together. Sorry. Yeah. Yeah.
Go ahead, Elmar. Okay, so
And
I was wondering if every zebrafish has a kind of different pattern of stripe by any chance, like as an identity, or is there any evolutionary reason to have a kind of those pattern mechanism for the stripes?
Yeah, so I mean, the patterning mechanism is the same across zebrafish, but interestingly, different strains of zebrafish, you can, I guess, you can like grow zebrafish to have different patterns. And they tend to be useful in like zebrafish work because they're good visual indicators of new zebrafish lines or different zebrafish lines. So like in the lab, we'll have like a specific zebrafish line that is more dotted than lined, right? And I wouldn't be able to actually tell you if they, like how related it is to a specific mutation, but it is a good visual indicator of different mutations and zebrafish lines that we have in the lab. But the patterning mechanism is consistent across. I assume there's just different initial conditions, and that's how we get those unique zebrafish patterns.
Thank you. Yeah, just I guess like those different patterns could be helpful for mating or something like that. Yeah, out of my imagination. Thank you.
That's a good question, actually. I can ask my advisor is Maria. I can ask her about, I guess, the value of stripes, because I don't actually know entirely the value of stripes, but I'm sure she will know. I can ask her and get back to you.
Great, thank you.
Okay, well, our last presentation will be CTrinity, JH, Elmar, and Jonathan.
So our model is about human pluripotent stem cell self-organization. Okay, so as a reminder, our project is based on Libby 20. Oh, wait a second. Yeah, Libby 2019 on the left, which the cellular pop model of stem cell self-organization using machine learning. The purpose of the model is to replicate the in vitro experiment in Libby 2018 on the right. The same author's previous wet lab paper about stem cell patterning. So we really appreciate the feedback from all of you, and this is kind of our response that we tried. So the feedback is like mainly categorized into 3 types. Like, is there any user-friendly exercise or explanation? So Jonathan has uploaded the NanoHub supporting document and video tutorial. And the second feedback is why not show more CC3D code instead of the Morpheus code. So Elmar will explain the difference between Morpheus and CC3D code for the model. And the last one is, what is the goal of this simulation, or what does it mean to manipulate those CDH1 and ROC1 molecules? So I will explain this part as follows. So the purpose of our project is, one, to replicate Libby 2019 model and translate the model from Morpheus environment to CC3D modeling environment to not only test the model's replicability but also the compatibility between two different environments. Also, for those who want to see the publication results but are inexperienced with cellular pot modeling, our application on NanoHub will be a nice user-friendly interface, getting hands-on experience in modifying the gene expression and see how they affect stem cell organization visually. So I will make this overview of biological background brief because we already went through this before. It all started with organoid, which is a 3D ball of self. This organoid, 3D ball of self-organized pluripotent stem cells. It is kind of a simplified version of organs, as you already know. So why do people want to use these organoids? Because you can study the complex biology of human organs, which you cannot do in animal models. And also it can be used for drug discovery in more humanized contexts. So whether this organoid will be brain organoid or liver organoid depends on its starting material, the stem cells. So what is pluripotent stem cell that we model? They are cells that have the potency to self-produce, like here, or differentiate into any types of cells in the body at the bottom. Then the question is, what makes them to self-renew or to commit to a particular cell type? Or what defines their cell fate? So there are a number of factors to play a role in stem cell differentiation, such as soluble cues like cytokines here, or, for example, autoinducer in quorum sensing, or physical factors including cell contact energy here, or stem cell niche, which means surrounding environment of the cells, although it is not here in the figure. So these many factors can contribute to the decision of the stem cell fate, like if it is gonna self-renewal or differentiate into some specific cell type. But the intracellular here or intercellular contact energy should be most relevant to our model, because our model is based on manipulating the expression level of two molecules, CDH1 and ROC1. As you can see, CDH1, these blue proteins, are the cell adhesion proteins regulating intercellular contact energy. And this ROC1, the red one, this protein regulates the contractile force of the cytoskeletal structure within a cell. So this is wet lab data from Libby 2018, manipulating gene expression of ROC1 and CDH1 using CRISPR interference technology, so that they can consequently control the corresponding protein amount. So the green means wild type cell, red means the cell type with ROC1 knockdown, and blue means cells with CDH1 knockdown. If you see the first row, there is no gene knockdown. So kind of they are in the homeostatic contact energy and tension status. On the second row, the red cells with ROC1 knockdown moved out toward the edge because those cells lost cytoskeletal tension within the cell, like rubber band, which lost elasticity and increased stiffness. On the last row, the blue cells with CDH1 knockdown lost adhesive contact with other cells, so they isolated themselves from others. So why the heck do they manipulate cellular behaviors like this? Again, this physical factor changes the cell's morphology and their niche and contributes to their cell fate. So we can say this manipulation kind of breaks the pluripotency of stem cells so that they can differentiate, the stem cells can differentiate into a particular cell type. It is great that we can control the stem cell behavior like this, but to control the gene knockdown timing, duration, and proportion, and to see some significant differences between the conditions, we would need to perform 675 total experiments with 13 biological replicates, according Libby 2019. So here comes the in silico model necessity to simulate all possible conditions to figure out what are the optimized parameters to manipulate these conditions. The cellular behaviors become simplified to reduce complex interactions into quantifiable relationships using space, time, and protein expression to develop the in silico model. The space-time relationships were used to represent velocity, while protein expression time relationships were captured with a temporal knockdown over a few days. Additionally, protein expression space relationships were shown by characterizing spatial patterns. Cellular POTS model, also known as GGH model, this was adopted to replicate the in vitro system in a 2D square lattice grid. Regarding machine learning, they use quad-tree image decomposition in combination with the Ripper-K rule induction algorithm to quantify the in vitro images. Using this method combined with particle swarm optimization, they made a CPM, or the cellular POP model, in silico experimental parameter sweep to find the optimized parameter settings for the in vitro pattern. So overall, the parameters we use, like which molecule to knock down, how strong to knock down them, when to knock down, and what ratio to take for seeding the wild type and those two knockdown cell types. All right. So before we move on to the app demonstration, let me briefly discuss the limitation or assumption of this model. First, this model assumes gene expression level correlates with protein level. Of course, as you can see in the figure on the right, the gene expression level, protein production starts at transcription from DNA to RNA here, and continues with translation from RNA to protein. But genome-wide correlation between expression levels of mRNA and protein is very poor. It's around 40%. So you cannot say because we knock down ROC1, ROC1 protein, because we knock down ROC1 gene, like for 20%, we cannot say the ROC1 protein is expressed like 20% less. We cannot exactly say that. Secondly, I have described the modification of these two molecules change the cell morphology and make the stem cell commit to a specific cell fate. But Libby 2018 already found that they are not enough to break the pluripotency. They measure the pluripotency transcription factors, and they found this kind of limitation. Because there are I already mentioned there are many factors to decide the fate of stem cells, which can be reflected in the model later, hopefully, possibly. So therefore, this model will be the first step to controlling the ultimate cell fate at the end of the day. Okay, take over, Jonathan.
Yeah, sure. So I'll share my screen real quick.
Okay.
So I'm going to give a quick demonstration on the app. First off, we'll just show that last slide here. And I'll show you this as well in the demonstration. But basically, there are two cell types. You can choose between the wild type, CDH, or ROC1. Two cell types will be in the simulation at the same time, though. And you can set the protein expression level for each. Actually, really just for CDH1. Wild type is going to be 100% at all times. ROC1 is going to be set at 20 default, but CDH1 you can set at different values. I'll show you some of the values and then which ones are have been yielding the best results. You can set the knockdown time as well, the number of cells to seed as well, and which plots to show. The defaults with adhesion and contact, however, there are a few others which can be set as well. Okay, so just to give you a look at the front page of the NanoHub application. So once you click on the link, it's not, my Google Chrome is really not cooperating with me today, but assuming that your browser cooperates, it will show you this page. We have the abstract. It's powered by our references, publications, all that kind of thing. And in a new edition that we've drafted but hasn't been published by NanoHub just yet, the abstract will also include a link to a YouTube video tutorial which has been posted and is in the document. So some other things to note on the main page here, you can click on the supporting docs, and that shows the exercises. Now, these exercises are just some very basic things that you can try, some variables that you can set. And yeah, Google Chrome still not working. Great, cool. But it's okay because I memorized some of the parameters that we can use. But basically, that document will show you a few different parameters that you can try, as well as suggesting some parameters and plots to look at on your own time for the user. It also gives a bit of a description on the fundamentals of the biology, what it all means. Okay, so when you open up the NanoHub app, I showed you this last time, but I'll just really briefly show it to you again. For those of you who have been using NanoHub this semester, this will be a little bit redundant, but you can click full screen. You can change the window size down here as I did. And you can tap on window here and tile all of the screens that are being shown. But before you get started, you're going to want to make sure that this play button is pressed. It defaults as being pressed, but it's going to be important for the simulation to begin once you make the settings in the steering panel. So here's our steering panel, and here are the parameters which I mentioned based on the slide, but I'll go into a little bit more detail on each. So there's a cell type 1 and cell type 2. What's worked out really well for us at getting the multi-island pattern in the publication has been setting wild type as the first cell type, and the second type is CDH1. So as we mentioned, wild type defaults to 100% expression. ROC1 defaults to 20% expression, which is why it's called ROC120 here. CDH1, you can pick any value between 0 and 100. So for example, in this case, we set cell type 2 to be CDH1. So you would go to the cell type 2 CDH express fraction and click on it here. You have a sliding bar you can choose the expression for. So in this case, we set it to 0 expression, which is the default that, well, actually in this case, we're going to set it to 50 just to show you how the expression shows up in the simulation. CDH1 with an expression of 0 can get a really good multi-island pattern, but CDH1 with an expression of 50 also gets a decent pattern. And then from here, you can set the knockdown time. This is the amount of time Monte Carlo steps in the simulation where that knockdown takes effect. We're going to have it start right from the beginning. There's going to be 50 cells of each. That's what the seed shows. CT1 seed is the number of cells seeded for cell type 1. CT2 shows the number of cells seeded for cell type 2. And then from here, these are just the plots that show. We're going to leave adhesion and contact on as the defaults, but say you wanted to look at how the volume changes over time, or the surface versus the volume, or just the plot of how the time changes, what the contacts look like, which we have default, or the velocity, you can set those here. And those are just on-offs. So whenever you're ready to get started, you change wait to go, and the simulation will begin. To get it to look, have them stacked all next to each other, you can click on tile and window there again. As you can see, the contacts are changing. The adhesion energy is changing. There's a legend here for cell contact evolution, which is kind of hard to see minimized, but you can click full screen and it'll show you. So, but let's take a closer look at this. And we'll leave it running for just a little bit. But as you can see in the blue, based on the legend over here, in the blue, the wild type is the blue cells that are appearing, and the CDH1 with an expression of 50 is in the green. If you want to zoom out the simulation, I suggest clicking the zoom out instead of scrolling to zoom out. If you scroll to zoom out, it's very touchy, and you'll all of a sudden be looking at the simulation from space. So zooming out with the zoom out button is usually the best way to go about it. So we're going to let this grow just a little bit, and I'll make some comments. So you may recall the first time that we presented this, there was this strange thing that was happening in the middle, where some cells were smaller than the cells on the outside. Elmar will explain this a little bit more in the code and the changes, but as you can see, that's not a problem anymore. The cells show up in similar sizes to each other in similar regions. There's no regions that have smaller cells than others. So as this is changing, you can notice some parts start to break off and become segmented. For example, we have a smaller island there. But as the simulation keeps running and keeps growing, slowly but surely, there will be islands. So let's see if I can load this once again. Oh, here we go. Okay, so some cell parameters to check. WT and CDH10 with knockdown time 0 ends up getting a really, really good multi-island. So the parameters that we have set now, we'll still get a decent multi-island pattern, but the best one was with different parameters. And then whenever you're ready to stop, you go back to the control panel, or the steering panel, excuse me. And change the weight. You can also stop the simulation up there. And then whenever you're ready to get started, just press play, change it back to go. So that's a bit on the simulation. I'm going to change the parameters and let it run with the best parameters throughout the rest of the presentation. And if you'd like, we can revisit this at the end.
Great, hope you can hear me. And I wait. Presentation B. Can you see this?
Yes.
Yes, great. Okay, so I'm gonna quickly talk a little bit about the source code, because we translated this Morpheus model to the Computational 3 model. I want to compare what we could do as they did and what there were slight discrepancies, slight changes that we had to take. So the basics, we had the same size of the lattice grid. It was 1.2 millimeters, square millimeters, and or 1.2 times 1.2 square millimeter. Then we had a neighborhood order of 4, so this was the same temperature of 10. In Morpheus, they have this yield 0.1 that seems to be the default, which somehow is for the membrane deformation resistance, and it's a kind of an offset to the Boltzmann constant. That's what they write, and it's referenced by a paper in Computational 3D. There's another possibility for that, so we just left it away. We don't think this is too much influence. Then there is the time unit that is in Morpheus by hour, and in CC3D we have strictly Monte Carlo steps. So what we did, because this hour are kind of neat when you compare to wet lab experiments, is that they had a doubling time for the cells, and we took this doubling time and thought about that there will be 1 pixel added per Monte Carlo step, and like this we could conclude that about 8 Monte Carlo steps are resembling 1 hour. It could as well be 9 or 10, but we settled on 8. So just didn't matter which cell you took to calculate this. So then they had random seedings. In Morpheus model, they always seed 100 cells, and you can define with how much you seed from one or the other cell type. This is one of the parameters that we have. So we implemented as well this random seeding, but we made like this, that you can choose how many cell you want for each of the cell type. It doesn't have to be to sum up to 100. And as well, we seed in the same matrix part from the lattice. So I stated, and this is the Python code that we wrote to do the seeding, and it's kind of neat. So you can set for even 0 cell of a type, and it will not break, and can even make both 0 if you want to. Then all the stuff will break. But and this nice loop that finds an empty spot, and there you place a cell that is in the beginning 4 pixels big, and it has this flip-flop mechanism that always sits one cell and the other cell, so that the cells that I not first seed all the cells from cell type 1, because then they have an advantage that they have all the spots free, and then the cell type 2, it's really always flipping between the two cell types. Yeah, so that's why maybe the code is a bit longer. Then cell cycle, we had defined here in Morpheus. So we have, this is similar to the fish group. So we have a known time for each cell that is kind of the generation time that the cell has. And this one is resetted when actually the cell enters divides. So this division time counter is resetted and starts to count again. And then these different cell types have different generation time. So it's just always compared with the cell counter to the cell generation time. And when it's higher than the generation time, then the cell splits. So this cell, unlike the ones that we often implemented, are not divided when the volume size is doubling. They divide when the time is ready. And in the beginning, every cell is by random seeding get a known starting point for when he divides next time. And this is, so I jumped a little bit around, but so this was how it's specified in the Morpheus model, and this is what we had to do in CC3D. And really, so it's in the steppables, and really in the beginning, we calculate these Monte Carlo steps with this factor from 1/8 back to hours. And then, as you know, from the presentation, we have this knockdown time that is earlier than the 0 time point. So we have to take the smallest of these knockdown times and add them to the current time point that we have, that we really know in which hourly time we are inside the experiment. And this part I explained before. This is the cell division and how this one is introduced. Sorry. So then we come to area and surface implementation. So mainly they had an area, that is the cell volume that they implemented, and they specified actually a lambda for the volume from 1 and the lambda from the surface for 0.5, which is very, very soft and weak. So we just took 3 for both of them in our implementation. And they, in Morpheus, I think it's the standard way, you don't specify directly the surface constraint, you just give a factor for that. And this is around 1.3 is this factor. So we did, James helped me to do this calculation the right way. This was actually in the last time when we showed the simulation was wrong. So more or less, you could do it on a circle, but you can do it on a square. So it's that you take the volume and you divide it by the square root, or you take the square root of the cube that you have, and then you multiply it by 4, and this gives you the length of the cell, so the surrounding, and then you multiply it with this 1.3, and this gives you your surface, the target surface that you set. And you could do the same with the formula for the circle, or you could as well use not the actual volume that you have from the cell, but the target volume from the cell. But in the end, we tried out all of these settings, and we settled on taking the cube and as well taking the actual volume of the cell. Then the velocity, that's very interesting. So this was the missing part. This was why our cells always were really having a lot of pressure in the middle and having these very, very small cells as soon the population was growing. And actually, there is an additional plot that you can see in the NanoHub simulation. There is as well the fields that you not only see the cell types, but you can as well see the pressure. And pressure was going really in the thousands. So, and it was because we didn't implement it actually this persistent motion the right way. So how it's done in Morpheus, they have no direction, so it's Brownian motion, totally random, and they have a decay time from 0.03 Monte Carlo steps, and the strength of this motion is set to 9. Now in CompuCell, you cannot do calculation in non-integers Monte Carlo steps, so 0.03 you have to calculate up to 1 Monte Carlo step, and this means that you end up with the force of 3,000. That's how strong the motion is from how the cell moves. And this that's what we said in there. And of course, because it's done each step, the cell is moving a longer distance than when it's done every 1,000th of a step. So we think as well, the cells are having not anymore this pressure coil in the middle with the small cells. So they spread out more, but because we only can do it each Monte Carlo stop, we think as well that's why the cell is, our colony is a little bit more fragmenting, and why you saw that there was one part a little bit breaking off. And according to the publication, they came up with these values from wet lab experiments, but just looking at the number 3,000, I think it's a very, very high value. But we took what they used, and that's what at the moment runs in NanoHub. And this is how we have to implement in CC3D. So we set 3,000 for the lambda. We set the persistence vector to 1 because it's per 1 MCS. And then we had to specify in a cell dictionary how we do this. And then this lambda vector XYZ is how you really set the direction. And this is how you set the angle. It's just a circle. Yeah, we did this actually in class. It's just the circle, and then you find randomly a value where you will walk next, and with sine and cosine, and then you set your lambda vectors. So this is how we implemented this velocity CC3D. Then contact energy and adhesion. So this was one thing that we have huge discrepancy to the code that we had originally in Morpheus. So I first explain how it's done in Computational, because I think we didn't do this really in class, and we think that this is the way that we understand with the Morpheus implementation. We had a little problem. So first of all, you can, if you do this adhesion flex implementation, you can actually calculate this back to what we are used to, the contact energy specification that the model sorts. But you cannot just take any contact energy quantification and just calculate it back to the adhesion flex. But the adhesion flex is more how you can think about it in biology. So as we said, we talked about these cadherin molecules, and you can think about how many, each cell type, how many molecules, what is the density of these molecules on the cell type, and that's what you think about. So when you think about the medium, we set this to 0 density. The medium doesn't have any of these cadherin molecules. And actually, we oriented us with the value that he gave here. We oriented us at the original Glazier paper from 1992 with the values that they used for cell sorting to be sure that it sorts. And Libby, the paper that we oriented us, and Morpheus, they set this value as well to 0. But now the strange thing compared to Libby happens. So we think about that in the original paper, they had the value of 14 constraint energy for the stronger one. There was a stronger and a weaker one. And for a default, we set the stronger one. And so, okay, the difference was actually 14. So the medium had actually the energy 16, and the cell type that was farther away from the medium had the stronger differences. That was set to 2. But later on, this as well, James explained me, they came to the conclusion that it would have been better to have actually negative adhesion energies for the cell types, because this makes in the end the cell independent. So the density from this cadherin molecule, it makes it independent from the length of the surface that you have. And if you have positive values like we usually have in our contact energy, then our contact energy is always directly dependent from as well the surface that we have. So it would be much better to have your negative default values, but okay, the values are as they are. But that was we used here the negative values because they make more sense to us, and these values are actually positive. But when you take the formula to calculate those values from this adhesion flex specification back to the contact energy values, then they become negative. Good. So then you can think about when there are two cells that are in touch with each other. So one cell have more cadherin molecule than the other one, and so the minimum will be the one that dictates how strong they stick together. And then you have this lambda factor, the kind of strength factor that brings the whole thing into relation from the whole model, and this one we set to 1. So now looking at the Levy model, when we overtook what they wrote, they had negative values. They had kind of like not cadherin molecules being there, what they said there. So their medium was 0, and the cells had negative cadherin molecules. So the most stickiest thing in their model was actually the medium and not the molecules. And to correct for that, somehow they didn't look, they took the mean, they took the max there. So somehow they had turned around the whole model in their implementation. So, and that's why we had to come up with re-implementing it from a new way. I think I said everything I want to. Yeah, there's one more thing I want to mention. So this density from 14, this difference from 14 that we know from the original paper and that we first took for seeding, this is an okay choice if you have a steady difference between medium and between your cell type or in between the cell types. But we have a temporal knockdown. The whole thing is that you temporarily knock down ROC1 or temporarily knock down CDH1, and over time, our knockdown cells will become more and more and more again like the wild type was. So they grow back to this wild type condition. So when we take here 14, then 14 will maybe will only be, the difference will only be 14 when there is really just the knockdown time, or time 0, or wherever the knockdown is. And to have a little bit more force, we doubled just the time, and we made the difference 28 to the media for the stronger cell, or for the strongest that we can reach. And then according how you set your parameter, according to the percentage that you take for the cadre knockdown, or the ROC, by ROC 20 it's set, by ROC 1 it's set how much this difference is. So there you can influence how much this gap really is, and this will in the end change the way how it sorts. Okay, so looking at the Morpheus model, as I said before, they had negative values. They had the max value that they used between these molecules. Yes, the max value is here specified. And then this was what is called the Morpheus homophilic adhesion. It's the same as this adhesion flex that I explained. So you see as well, you have the cell type to a medium adhesion that they specify, then the cadherin molecules in between one cell type or in between the two different cell types or the other cell type. But it's actually resulting in the same, as I told before, you can do the same. And then as well, they had another strange setting. So you might remember that the general setting was that the neighborhood was 4. But for this surface energy, they set this neighborhood to 1. And actually, we looked at this implementation and we played around with it. And the whole thing is that the cells behave, they're like magnets that are same poled. So they float on top of each other. Like you maybe have a kid played as well with magnets. We did it at least. And when you give enough pressure, so when there was pressure in the colony was growing, or when they had the pressure from the movement, from the force, from the vector, then there is enough that the cells were touching each other. But it was really like a kind of this same pulled weak magnet model that they had their organoid modeled, which I believe, I know the model works because we could run it in the Docker container and we could get results. But I think that they were not totally aware what they have modeled there. It just somehow worked. That's what I think. So yeah, that was how we translate this implementation. Then the last part I already talked about a little bit. It's when you come from a knockdown cell back to your wild type cell. This was implemented in the Hill response function, or at least that's what they claimed in the paper. And they had the Hill power from 5 that they specified. And it was this is the Morpheus model. Now it's on the lower button, and it was a quite complicated if then else construct. So more or less this, if this condition, they had as well kind of a burning time, then that, so, and then if, and there was quite some expression, then that, and else. So they had about 3 or 4 conditions that they had. And when you really looked at it and how they regulated, it was as well the proper Hill function that they wrote there. So we as well simplified this one, and we had just an if else, and we couldn't really totally understand what they tried to to describe with this formula. So we as well, we wrote it then back into a simple function. And this hill response function is the volume constraint that influences the surface constraint, the adhesion, and the generation time. They are different for each of the cell types. So this was how we finalized our whole translation from studying this Morpheus model and transforming it into CCCD. I think the final model really looks similar to what we saw in the real model, but quite similar, as good as it gets. And yeah, we think we have have our task fulfilled and have translated this model. We had future plan, which actually is vacation, and yeah. So if you have questions, please let us know. Yeah. Oh, have I lost everyone? I don't know.
Please go ahead with questions. I just want to put up a thank you slide because everybody did a great job. So,
Thank you.
and you could reshare if you need to show something, Elmar and
Yeah.
James. So just let me know. Go ahead, questions.
So in the end, the weirdness in the center was just lack of movement.
Yes, it was that we. I didn't understood. Yes, how to translate these movements that I had that were these 0.03 Monte Carlo steps into the right way. I made one Monte Carlo step and I had the strength by 9, and this is no power compared to 3000, right? So that was yes. But then there were in the first model that we had last presentation, there were some other problems that James helped me to resolve. So one was that my surface calculation was wrong, as coming from the volume to the surface calculation, and he helped me that one. And then he explained me as well that we should go with negative, or that the right way to go would be with negative adhesion energies, so that I got an adhesion model running that made sense, because I think the original Morpheus model didn't make sense, and the one that I implemented was as well not totally correct. So there really James helped me to get those two right. Yeah, and thank you very much for that.
Well, I talked to them last week. I talked to Lutz and Jörn last week a bit, and one of the things that there are two things. There are many, but there are two which you ran into. One of which I put in the chat. One of which is that they automatically rescale the contact energies depending on the interaction range. And so if you have an interaction range of 1, they divide by 2. If its interaction range is 2, they divide by 3 or something like this. 3, they divide by 6, and 4, they divide by something like 10. And so there's a scaling factor between the values of contact energy in their code and ours. And it's possible to look it up. The exact numbers are in a paper written by Stan Murray, who doesn't use Morpheus. So it makes it a little harder to find. The other thing is that for surface area, they always calculate the surface area. We always calculate nearest neighbor surface area. They calculate the surface area using the same interaction range as the contact energy. And so the surface returned by their surface measurement is bigger than ours for the same simulation. And so there are a bunch of hidden. Computational doesn't do automatic rescalings. It would be nice maybe if it did, but the problem is that when you do automatic rescalings, it's not always obvious what those rescalings were. And so I think some of the problems came about because Morpheus is being smart at trying to guess what you want to do. And when it works, it's great. But when you're trying to translate it out, it's not so clear. So I'm very impressed that you were able to get this all to work. I think it was a big effort, I know, for everybody. Other
Yeah,
questions?
thank you for all the help as well.
Any other questions, comments? Okay, well, we're a little bit over time. I thank everybody for sticking around. And they were great presentations. I really appreciate everybody putting in all this hard work. It was really, I'm very impressed by what people did. And I do know it's a lot of time and effort. And I hope people felt they learned something. And I do appreciate you sticking the course out and doing such great projects. So that's officially the end of the class. If people want to talk to me, Giuliano, Hayden about trying to update models, improve them in ways, we're always available to try to do that. But I do appreciate everyone's work. Thanks so much. Good night.
Thank you.
Thank you.
Thank you.
As well.
I had a quick